All right, are you all set? Yeah. All right, thank you. All right, everybody, we're gonna go ahead and get started with day 3. Again, we're all excited to be back here, CNRA headquarters building. This is the only time, just a heads up, that we do have a concurrent thing going on. The cyanobacterial harmful algal bloom sub-team is meeting upstairs from 8:30 to 1. So again, the bathrooms underneath the stairs and around the corner, Wi-Fi, CNRA guest, no food or drink in the auditorium. Thank you ICF for sponsoring the coffee. If there are any accommodations needed today, speak with Christine at the front desk. If you're online, feel free to drop a question in the Q&A. And a reminder that posters are due to be taken down no later than 2. If they're still up after 2, apparently CDFW will bring them back to their office and you can get them if you want later, but try and take them down by 2 o'clock. And with that, we will roll right into the session 11.
All right, thank you, Adam. My name is Brian Hutchinson. I'm with the U.S. Fish and Wildlife Service, and thank you guys for joining for session 11. We're going to be talking about food webs and predation, and our first presenter today is going to be Lillian McCormick.
Thanks so much, Brian, and thanks to everybody for showing up on the early morning time on the last day of the workshop to folks in person and online. So my name is Lillian, and I'm going to talk to you today about food web interactions. First, I'd like to thank my co-authors as well as the groups that have sponsored this research. So this has started when I was a postdoc with California Sea Grant and the Delta Independent Science Board, and then continued on at Bureau of Reclamation. So we'll jump in. So many agencies, as you all know, are interested in improving life cycle models and decision support tools for smelt, salmon, and many other listed species. And so there's been a recent interest in another set of interest in further understanding food web dynamics. And I've listed some recent works that have incorporated food web knowledge, but really the larger ability to listen to current projects will be in this session. So stay tuned for some of those projects. But I will say that understanding species interactions, especially the biotic part of those species interactions, can be pretty complicated. And the combined heterogeneity of the Bay Delta estuary and the potential for siloscience can also serve as barriers. So before starting, I want to say that in part due to IEP research and all the research that you all have contributed, fortunately, the Bay Delta estuary is a really well-studied system and has an excellent history of studying food webs. But we do know that food webs have changed over time in this estuary, primarily due to many physical changes, the introduction of non-native species, and also climate change. So here on the left, you can see a figure of the number of publications that are about food web interactions over time. And this is just showing a snapshot of this between 1990 and 2023. And so what you can see is this really large increase in the number of studies that consider food web interactions in about the early 2000s, which was empirically when the pelagic organism decline was detected. So you've probably heard many other folks talk about this, but this was a regime shift marked by a decline in 4 different fish populations, delta smelt, longfin smelt, threadfin shad, and striped bass. And it was largely attributed to predator-prey relationships, increased water exports, changes to abiotic conditions such as temperature, and also the effects of non-native species like Potamocorbula on water clarity and food supply. So with all of this food web knowledge, I was really interested in understanding the spread of science that's been conducted in this region, and just to see whether we could identify any science gaps. So for example, what types of species do these studies focus on? And what is kind of the spatio-temporal spread? So are specific regions focusing on specific species, or does it capture the heterogeneity of the system? And then I really wanted to take a deep dive into the species interactions and see what types of interactions were primarily studied and understand whether there were any missing connections, particularly that would be relevant for management. So not surprisingly, the tool for the job was a large structured literature review. I didn't realize how large it was going to be until I started. So I used two search engines, Web of Science and Google Scholar, with fixed search strings. Restricting from 1990 to 2023 because I just needed some brackets because there is so much research about food webs. So after some document category exclusions and removing duplicates, I started my initial screening of about a little over 1,200 papers. So this is still evolving. So the results that I'll present today are just a snapshot of what is contained in this large database of species interactions. So I'm definitely open to comments and stuff about what other things may be interesting to consider. So just to follow along with this diagram, this is a prism diagram showing the flow of paper inclusion and exclusion. And so just to summarize, there were a little over 1,700 papers that were considered. Again, about 1,200 that were initially screened. I went through full text screening for about 488 papers, and then the results will present from 278 papers that were considered to be kept for throughout all this inclusion criteria. So today I'm going to present two types of results just because I can only present so much. So first, I'm going to talk a little bit about the regional variation in the studies, what they study. So the types of species, the status of the species. So whether they're listed species, native but not listed, non-native species, or multiple species status, and also the trophic level of interest of the study. And then second, I really want to try to understand how these data can be used in management. So understand the beneficial uses of these data. So first I'll talk about the regional variation. I'll first walk you through this plot. So this is a chord diagram that shows directional species interactions. So you'll see the circle with lots of different colors. Those colors represent species groups or species categories, with the color indicating the first species in the interaction. So for example, you can see, I don't have a pointer, but you can follow along with your eyes. So if you look at any of the pink lines and moving over to the blue section, that's indicating bird predation on fish. So you can follow the lines to kind of understand the species interactions. Okay, so now that you understand the plot, we'll go over a little bit of the results. So studies that, and before I start on that, I'll just mention that the graph on the right is showing the specific regions that I broke things into. So San Francisco Bay, Sassoon, the Delta, and then rivers. And those are all highlighted in the different colors. So for studies that were conducted in San Francisco Bay, they primarily covered native species and secondary consumer trophic levels. So as you can see by all the colors in this circle diagram, there were lots of variety of species that were studied, including many studies on bird predation on fish, and also mammal and marine mammal predation, as well as native versus non-native plant communities. For Sassoon, so we're going to move eastward now. Studies that were conducted in Sassoon primarily covered multiple species and also lower trophic levels, such as primary producers. There was an increase in the number of studies, interactions involving fish, predation on crustaceans and copepods, and also understanding what copepods eat as well. So birds and mammals were still studied, but there was a switch to understanding mammalian predation on birds rather than bird predation on fish, as there was in San Francisco Bay. So moving eastward again to the Delta, studies conducted in the Delta were primarily non-native species and also secondary consumers or multiple trophic levels. And a lot of these studies covered non-native species or kind of multiple status. So looking at both non-native and listed species. So you can see that there's a big change in colors here. So a lot of these studies were very fish-centric, but we start seeing some different prey items for fish, including insects, amphipods, copepods, and crustaceans. And then for the rivers, this is even more fish-centric, but riverine studies focus primarily on listed species and also secondary consumers. So again, lots of studies on fish, but now it's kind of shifting to fish predation on insects and fewer studies about fish predation on crustaceans and copepods. Birds became more commonly studied again, but this is likely due to studies of rivers around San Francisco Bay, such as the Petaluma River. So you can really tell that birds were studied in San Francisco Bay. Okay, so since other folks in this session are largely going to be covering food web interactions with salmonids, not excluding WIM, talking about copepods, I thought I would use another famous fish as an example. So this is a chart of species interactions as discovered by this interactions analysis with Delta smelt. So in these charts, I'm going to be showing you a couple of these. The circles or nodes represent species or groups of species, and the lines show connections between species, with the width of the line indicating the number of studies that detailed that interaction. The colors, similar to the other plot, the colors are representative of the first species or group in that interaction. So for example, this dark blue color with LMB in there is showing largemouth bass predation on Delta smelt. So you'll see that I also included multiple life stages of Delta smelt. So this light teal up in kind of the upper right quadrant of the plot is showing Delta smelt eggs and larvae or early life stages, ELS. And then the kind of darker teal in the center is showing juvenile and adult Delta smelt life stages. And I think that's mostly, I did forget to mention that the types of lines, so whether it's a solid line or a dashed line, indicates the type of interaction that is being studied. And that is in the key on the bottom right of the legend. So this is a lot. How can we understand how this all can be used for management and help us understand whether there are any connections that are detailed in here that are not currently being used in management? Well, one of the tools, obviously, that we use in management for listed species are models, and that's really important for us. And that's how we in part understand how species respond to either human stressors or just different changes in environmental conditions. So one of the models that is used in the management of Delta smelt is the individual based life cycle model in R, or IBMR. So here's a diagram showing some of the details of this model, like the inputs and the outputs and the different types of data that are incorporated into this model. So I'm not going to go into specifics of this model, but I do want to show some of the food web interactions and highlight some of those sections within the model, such as the prey items for Delta smelt, as well as this kind of mortality box, as well as some maybe indirect inclusion of food web dynamics, such as consumption rates or prey density or prey vulnerability that are included in the model. And I also do want to say that this is not a criticism on this model. This is merely being used for comparison so that we can understand how these species interactions can help us understand what might be able to be added in certain management tools. So this is not a criticism on the IBMR. So how does this complicated plot compare to the IBMR? So let's start with prey items. So now I've highlighted the different prey interactions where Delta smelt is preying on something. And again, you can see the key on the right if you have trouble understanding some of this figure. So let's talk about what's included in the IBMR. So these are the 12 prey items for Delta smelt that are included in this model. And I will kind of walk through really quick to see, to compare across this figure and the model. So we first have Eurytemera and Pseudodiaptomus. Those are included in some of the orange bubbles on the right. We also have Acarciella and other calanoid copepods. We have other families of copepods, so Limnoithona. We have Copepodnoplii, which are actually already included in some of these individual species little circles. We have cladocerans like Daphnia species, and that's included in the cladoceran bubble, kind of in the center right side of the plot. And then we have all other taxa. It's a little hard to compare and see whether we can include all of that, these interactions. But if you notice in this plot, there are some other interactions. So for example, crustacean parts or chiropid amphipods or mysids. So that may be included. So what we see is really, really good we see really, really good comparisons between the prey items that are included in the IBMR and then also this plot. So moving right along to talk about predators, this is a little bit different because predators are not directly included with individual species in the IBMR. So this is a little bit different. So, but predation is incorporated into the mortality term, which includes predation in the South Delta or other areas, and also entrainment. It's also indirectly included in the model because turbidity is included in the model. And we know that when turbidity decreases, the risk of predation for Delta smelt increases. So, but if there was a situation where this model or other models wanted to include individual species predation on Delta smelt into the model, they could look at this interactions analysis to figure out which species may be more important to include. And then finally, with the species interactions, one of the cool things about it is that I'm not just looking at predator-prey interactions, but kind of some of these other interactions. So you can again see some of these different line types that are not just solid, but dashed or dot dash. And what you can see is that there are a whole bunch of species or groups that also interact with Delta smelt in different ways. So for example, some of them have potential for direct inclusion into these models, such as the overbite clam. So either from competition with prey items or direct predation on larval individuals. Or we can also have indirect inclusion to the model, again, similar to what we were talking about with the turbidity term, which we know that submerged aquatic vegetation and floating aquatic vegetation, SAV and FAV, also typically reduce turbidity, which can, again, increase predation risk for Delta smelt. So just different things to consider. Okay, so to summarize, I showed some regional differences in the species that were studied with the spatial gradients of studying native, non-native, and multiple species. So I will point out that this is definitely partially due to the heterogeneity of what species occur where in this region. However, this could be also due to potential bias in certain areas and regions and the types of species that they study. So for example, we do have birds in the Delta, but they aren't often studied with predation interactions. And so, but they're very often studied in San Francisco Bay. So that's just one example of this kind of regional bias that we do see within this research. And if you need a reminder of how different each region is, I've included the circle diagrams. I know you can't read the labels, but you can see hopefully some of the differences in color that indicate the different species interactions that are covered in each of these regions. And then we talked about how interactions could potentially be used for models that are used in management. So for example, there could be an optional expansion of some of these models or development of new models as new data becomes available. And also you could use this interactions analysis for troubleshooting models that maybe don't perform as expected, and that may be due to some of these missed species interactions connections. So finally, I really want to make this a community tool. So the whole design of this project was to create some sort of Our Shiny app or online tool where folks could input their species of interest or their region of interest and understand the species interactions that occurred surrounding that species. So, and I'd also like to have it be a tool that could be expanded upon by the community so that it doesn't just become obsolete in time. So right now the limit is 2023. I know there's more research that's been conducted since 2023, but there could be forms where folks could input the species interactions from their studies or from other studies that they're familiar with. And so this tool is still evolving. And so I'm still in process of making this. So if you do have ideas and comments, please don't hesitate to come up to me and chat about them. I'd love to chat. And thank you. I'll take any questions.
Thank you, Lily. We have time for quite a few questions. So if anybody has some.
Rosie.
Thank you. Really cool to see how this is coming along. This might be a too specific question, but I noticed in your chord diagrams, which you had said was, a line from one taxa to another was this taxa predating on that taxa. Is it always predation?
No, so that's a really good point, and I should have specified that. So those are just detailing regular interactions. So it's not necessarily like an exploitation type reaction. It could be, a beneficial interaction as well. Good
Cool,
question.
thanks, because I saw plants going to fish, and I got a little nervous.
How would that work? Yes, good catch. Thanks, Rosie.
Yeah, thank you, Lillian. That's really good, really cool. I particularly like your talk about bias because I've been complaining about this for decades, about the Delta-centric bias in this community and especially in management, but it leaks into the science as well. Much easier to get stuff funded, or at least it was in the Delta proper, or maybe Suisun Bay. And there's been a sort of leakage of terminology from the Delta into Suisun Bay and even further. So I wonder, have you looked at the bias from that perspective in terms of, for example, okay, longfin smelt, you know, 3/4 of the habitat of longfin smelt is invisible to the monitoring programs. You know, the larval part of it is okay, early juveniles, and then the adults. And, you know, so spawning, eggs, where are they? Ocean residents, we know they go out there. We don't know where, we don't know how long. You know, there's some huge gaps there. And so, but this is systemic. I'm just using that as an example. It's familiar to me because we're working on that. But I wonder what you can say about that.
Yeah, so I mean, that all makes a lot of sense to me after going through all this, because you definitely can see a ton of bias in the research. And so one of the things that I was originally really interested in was connecting research across these regions. So for example, you know, looking at studies that cover both the Pacific and other regions, so that you can kind of have more of a holistic view of what the food web is rather than just a small snapshot in one particular location. Obviously, research has its limits, and that's very understandable. But yeah, there is a ton of bias within the system, and I'm hoping that I can kind of dive further into these results to not only just look at, you know, individual species, but potentially look at different life stages, like you mentioned, so that you can really pinpoint different areas of science that could be studied in the future. So yeah, thanks. Oh, no, I know.
All right, we have time for probably one more. Take one online.
Yeah, we've got a quick one. From Laura, very interesting talk, Lillian. Did you show Delta smelt supplementation somewhere on the model, or is that purple dotted line from humans to smelt?
Yeah, so that's included in that dotted line from humans to smelt. So I did try to include human-centric interactions like supplementation and also the projects and any sort of entrainment that might be happening. So I did include human interactions with that. And I do remember reading about one paper with Delta smelt supplementation. So yeah, great question. And all of that was, I did try to incorporate all that into the analysis.
Thank you again.
Thanks.
All right, so next we have Elizabeth Greenheck of George Mason University.
Hi, thanks everyone for being here today. Today I'm going to discuss a project that myself and my PI, Reed Nelson, are working on, along with the folks at UC Santa Cruz NOAA. Cyril and Brendan are going to be here today, or they're here now. Oh, hi, there you are. Hello. So today we're going to be talking about quantifying component mortality rates of juvenile steelhead trout in the Central Valley. So we're going to focus on not just knowing how many fish survive, but why those fish are dying. So steelhead are federally threatened in the Central Valley, and they're distributed among the Sacramento and San Joaquin rivers. I'm going to be focusing on the San Joaquin for today's talk. And most people in this room are familiar with the population declines that steelhead and other salmonid have faced across their whole range. So this includes habitat alteration. Lillian talked about that with Delta smelt, where reduced flow makes these juveniles more visible to predators. So same situation with steelhead as they are out migrating. Also, the concern of invasive species, especially bass, in these systems that have been kind of thought to increase predation of these migrating salmon, and then climate change effects. So temperature being a big one, that's of concern. So for that reason, survival of outmigrating juveniles has been a concern across these study areas by a lot of people, a lot who are in this room today. And in the San Joaquin, in the area that we focused on for this study, survival was estimated to be between 15 to 54% from a study using acoustic telemetry from 2010 to 2016. In this study, they use traditional telemetry methods. So those tags get put into the system, and as they swim by a receiver, it tells you what fish was detected, where it was detected, and when. And so in order to understand whether a fish survived or was predated on or died from other causes, you have to infer their final states, either using stationary tags, so looking at a fish that kind of stalls out at a certain location, tag removal from the system, so a fish just going missing, they're not getting fished by humans, so we know that something else pulled it out of the system. And then other metrics that have been used are predator filters. So the figure to my right is from Buchanan and Whitlock 2022, where they looked at a lot of behavioral metrics of fish moving through the system and were able to identify smolt versus predator behavior to try to see how many fish were getting predated on. These studies used a single source of mortality, so just survival versus mortality, because they weren't really able to quantitatively or with a lot of precision actually parse out that mortality as predation versus maybe a water quality issue or that sort of thing. So with the advances in technology, acoustic telemetry has also advanced, and Innovacy, which is one of the companies that makes acoustic tags, has developed a predation detection acoustic tag, which I'll be calling PDATs, which are similar to traditional telemetry metrics. They give you the who, when, and where, but they also have a couple additional sensors. So these tags have a detection sensor or a predation sensor that is activated by an inert polymer covering. So on the figure to my right are a couple different sizes of these PDAT tags. That white film looking thing is that polymer covering. And when that polymer interacts with stomach acid, it will break down and trigger the sensor to let you know that your fish was predated. So these are obviously really useful in actually confirming predation is occurring rather than, inferring it from behavior. So briefly, how these tags work: once you tag your study species of interest and release it into the system, the tag will start recording the detection sensor. So if a fish is alive, this will be 1, and then it'll also start recording the body temperature of your species. So this is going to be ambient water temperature. If a fish is predated at time equals 2, that detection sensor D will flip to start recording the number of hours since predation, and then we'll start recording the body temperature of the predator. So if your fish was eaten by another fish, this is still going to equal ambient water temperature. But if a bird or mammal ate your fish, this is going to equal higher than ambient water temperature. So you're able to actually parse out additional sources of mortality, not just what's assumed to be, fish aquatic predation. And then as time goes on, that predation sensor will continue to increase in the number of hours since predation. And this allows you to back calculate when predation actually occurred if you have any gaps in your detection history. So these tags are pretty useful. So we were lucky enough to actually tag some steelhead. And in the study, we were able to partition juvenile steelhead mortality among its component sources. So we used passive and active telemetry, which I'll talk about in my method slides in a second, and then took these telemetry data and ran them through some Bayesian multi-state models to quantify component mortality. This study, we can have a second part to it, is assessing the utility of these PDATs when compared to the metrics that have been used previously. So that predator filter, looking at stationary tags. These tags are pretty expensive, so your cost of actually implementing this sort of study increases. So we want to see, how much better we're able to partition mortality using these. I won't talk about that today, though. So we released 300 hatchery-raised fish at Durham Ferry, which is shown on the figure to my right in the star, and that depicts our study area. So the San Joaquin River before it hits the Old River. We released 100 fish per month across March, April, and May of 2024. And all of these fish were internally tagged with PDATs with a 50-day battery life. We detected telemetered steelhead using 34 total receivers that comprised 17 receiver gates, so 2 receivers per gate. And each gate was about 1 to 1.5 kilometers apart. And then we supplemented our passive telemetry with manual tracking for 8 days following each release of tagged steelhead. During this time, we also recorded any predators that we saw in the system and then any habitat attributes such as oxbows, snags, those sort of things that might affect our detection probability. We also weren't quite sure what the actual delay would be for that predator sensor. So we decided to feed some striped bass caught by FISHBIO a little snack of a tagged steelhead. So we force-fed 11 striped bass in our study and released them into our study system with steelhead that had that PDAT tag. Of those striped bass, 9 were detected in our study system, and of the 9, 5 had a trigger delay of about 1 day. 4 fish did not trigger. We think this is because these striped bass left the system pretty quickly, so we just didn't detect them long enough to actually see that predation sensor trigger. But 1 day was what we decided to go with when back calculating the time of predation for our actual steelhead in the system. So with our manual and active, or our manual and passive telemetry data, we tidied all of those and created capture histories for each fish across the 50 days in which the tag should have been alive. We assumed that all fish were released alive in our study system. We also then created a second period, which comprised manual and active tracking because our detection probability was increased during that period. We had increased effort. And then our third period was our final state. So the remainder of the 50 days left in the study, were we able to see, you know, like what the final fate of these fish were. I'll note fish were censored after they successfully emigrated out of our study system, so they were not modeled after they actually left. So we just modeled fish that were within our study system. Breaking the data into these 3 periods also accounted for a decrease in sample size across our study, which you can see on the figure to my right for all 3 release groups. So obviously as time went on and fish emigrated out or were predated on, we decreased our sample size. We then took these telemetry data and applied a Bayesian multi-state model with 3 states for at least what I'm presenting today. So any fish that were alive in the system at the end of the study period or emigrated were had a true state of alive. Any fish where the PDAT actually triggered were determined to be aquatic predation. And then any unobserved fish, any stationary tags, we had about 4 stationary tags in our study, were assigned to other mortality. And the figure to my right shows the tracks of these fish in the study area, so you can kind of see what those data look like. So moving into our results. The numbers that I have to the left are overall for all 3 release groups, but the figure to my right does show component mortality within each release group. So I'll kind of iterate through this. So our study had really high survival. We had 78% discrete survival with a 71 to 83% credible interval. So that's in the yellow box plots to my right. We saw 15% discrete aquatic predation mortality with an 11 to 21% credible interval in the purple. And then we had 7% discrete other mortality in the pink with a 4 to 12% credible interval. I'll point your attention to the differences among release groups. So in our first and second release groups, we saw higher aquatic predation relative to other mortality sources. But in our third release group, we had higher other mortality relative to aquatic predation. And I'll talk about that in a couple slides here. For those interested, this is our detection probability across the 8 periods that were modeled. So we modeled daily mortality and then combined it for a final discrete value. And I'll note in our first release group, we had relatively low detection probability compared to the second and third release group. And that was a function of emigration time in the system. I have a figure in a couple slides about that, but for those interested, that is our detection probability. So to discuss kind of overall survival, we saw 78% survival in 2024. And when compared to the study from 2010 to 2016, this was a good deal higher. They had less than 50% survival in the same study area across all years of their study, except for in 2011, where they saw 54% survival. And we believe that increased survival in our study was a result of higher flow conditions in 2024. So the figure to my right depicts flow across our study area from a USGS gauge. The greenish yellow line in the background is that 2011 year with 54% survival. And then the red is flow during the 2024 study where we had 78% survival. We're trying to actually incorporate flow data into these models so that we can quantitatively assess this, but this is kind of what we're thinking is going on in terms of our high survival in 2024. I mentioned before that emigration time was different among our release groups. So our first release group had significantly longer time in the system relative to our second and third release groups, which emigrated pretty quickly out of the system. So when I talked about detection probability in that first release group being fairly poor, we believe this is a reason as to why a lot of these fish looked like they would have been dead, and then they really quickly emigrated out and the PDAT sensor never triggered. So they were just kind of hanging out for a long time. This also got me thinking about looking into other factors that may have influenced fish remaining in the system, like zooplankton abundance, other aspects. So we're trying to also quantify that by incorporating some of those data into the model. I mentioned before that our third release group had higher other mortality relative to aquatic predation. We think this could be a result of increased temperature. Obviously, our third release group was our latest release group. And you can see on the figure to my right, the yellow is that third release group and the temperatures that the fish experienced in the circles, because again, these tags record body temperature of the fish. And then the line is USGS data, which matches pretty well as we would expect. So you can see this third release group experienced higher temperatures, also lower flow. So we think we're going to try to, parse that out and actually include flow and temperature data into the model to quantitatively assess this. Okay, so getting into future work, we did actually observe other mortality in our model. So we're considering having a 4 observation and 3 state model where we still are modeling survival, aquatic predation, and mortality, but we're able to actually better, parse out other mortality for instances where we did actually see those fish die from stationary tags. So that's one model that we're still working on. I talked about incorporating environmental data into the model. That's something we're actively working on in both among release groups, so kind of a more easy model fit, and then actually indexing individual fish at time T data into the model, which is a lot more complicated and it may or may not work due to error and other things. I'll point out we did have 2 fish get predated on by an endothermic predator. So that's the top figure here. 2 out of 300 is not enough to assign an entire state to that predation. So we'll just have to qualitatively discuss that, but pretty cool to see. Like I mentioned before, we are hoping to assess the utility of PDATs as well as active tracking in assigning fates and trying to compare the component mortality we got from this study and then kind of removing all of our fancy tag information and running the models again to see how that parses out. For those that are interested in doing a study like this, we definitely recommend testing the predation trigger by force feeding some predators in your system. That was really useful for actually being able to back calculate when these fish were predated. However, we would also recommend maybe tossing a couple of those expensive PDATs just into the water to see how that polymer breaks down in ambient water temperature. That's not something we did. So there's a little bit of uncertainty with a couple fish, and we're not sure if the tag just broke down or what's going on there. So would recommend that if anyone's looking at using PDATs in their own work. Okay, so with that, I'd like to thank the funding sources, as well as the folks at FISHBIO again for getting us those striped bass, the folks at the hatchery, and then University of California, Davis as well for support with this study. And I can take any questions.
Yeah, thanks. That was great. I'm curious to, since you had those seeded fish, and it indicated that there were a few fish that you think were predated on, but they left the system too quickly, before you could detect the polymer breakdown. I wonder how that might potentially bias your survival rates, right? So, bias
Yeah.
them high. And I don't know if I'm sure there's a way to account for it. I don't know if you've thought about that.
I have a note in my R script that is like, hey, look at that again.
Yeah.
So we have, yeah, so a lot of the fish, especially in that second and third release group, left the system in like under a day. So it's possible that they were consumed by a striped bass, and that detection track is just the striped bass with the fish in it leaving the system in under a day. So yeah, it's definitely something that we could, I'm trying to think of how we'd probably, yeah, might have to create like another state for those fish specifically. It's definitely something we've talked about. Quantifying it in the model becomes like a whole nother issue, but yeah.
Prior or something?
Yeah,
Yeah, it's like an
definitely.
informative prior possibly. I don't know exactly how to do it either. I was just curious if you.
yeah, I've got a little note for myself for later. Yeah.
We have time for about one more question if there are any.
How did your different timing for outmigration and relief compare to the natural outmigration timing for steelhead in the system?
I would assume for this study, it was pretty similar to other studies. This is my first time working with salmon in this area. So I haven't run other data to see if this is on par with other studies that have looked at outmigration timing. But yeah, I don't know if probably a lot of people in this room would know better than me if this is on par or not, but I would assume so. Especially in a higher flow condition, those fish probably got flushed out pretty quickly. But yeah.
Pretty quick question. So we got time for one more.
Oh, cool.
I think you had it up first.
This is more, excuse me, more on the technical side of it. The PDAT tags, you're suggesting, or you're telling us that they transmit the temperature that's embedded within the tag code. And we have this endothermic predation, suggesting 40 degrees C or whatever it is. How is that functioning when it's a hydroacoustic signal if a bird, for example, predates on the species and it is out of the water, that tag code can't penetrate the water?
Yeah, it's not getting picked up at all. So that would be a really cool new tag where it's a combined radio acoustic tag. I don't know how you would transmit temperature in a radio tag. I'm not familiar with that technology, but unless a fish is in the system, we do not detect it. So
Right. So
yeah.
So if a bird were predated on it, I just don't know how we get a 40-degree temperature signal to.
Yeah, and it very well could be in like a shallow area that's getting hit by a lot of sun.
Sure.
I need to look into, there's not actually a lot written in like the PDAT info guide or whatever about like if if it chooses a temperature and sticks with it or what. So yeah.
I've worked with Nova Sea a lot over the years, and I've always had those interesting questions for them.
Yeah. Yeah, for sure. Thank you.
Thank you, Elizabeth.
Thanks.
All right, so next up, we have Jordan Massie with the Vermont, University of Vermont. Thank you.
Thank you, Brian. Appreciate it. And I think this is a nice follow-up, a great talk, Elizabeth. It's good to see some empirical data about predation. That's really exciting. Today, I'm going to be talking on a similar topic about predation and juvenile salmonids, using a statistical approach to better understand how the environment could potentially lead to some differential predation sensitivity among different ecotypes. So I want to start out by saying that this is a huge project that I'm presenting on, but this is kind of a collaborative effort with many, many different folks. So we're drawing on data that's been collected over about a decade by folks who have been working throughout the Delta and throughout the Sacramento River, including Cyril, who's here today, and his team down at UC Santa Cruz. So while I'm presenting and doing some of these analyses, this is really a large effort that includes many, many people. So here in the Central Valley, most of us are familiar with the plight of the Pacific salmon. And over the last couple of days, there's been some great talks that I've pointed out many of these different aspects of how we've gotten into the situation of decline. Here, I'll just kind of mention, the 4-H's. These are big things that have kind of left us in a situation where the populations have been diminished from their historic abundance. So we're dealing with habitat loss and degradation throughout the Sacramento, San Joaquin systems, still dealing with the legacy effect of gold mining and alteration, increased urbanization, revetment and channelization, disconnectivity from the floodplains, along with the legacy effects of intensive harvest that date back over 100 years ago, which have greatly depleted these stocks. That combined with the altered flow regimes resulting from the dams and water diversion really have altered the habitat and the dynamics of this system. And large-scale hatchery supplementation has potentially diluted some of these genetics, which have adapted these fish to be able to survive in these systems. So in the Central Valley specifically, there's been a massive body of research which has illustrated that the smolts, these outmigrating juveniles, may be a particularly sensitive life stage for supporting these populations over time. And in the Central Valley, survival of smolts has been shown to be much lower than some of the northern rivers, like the Fraser up in British Columbia and the Columbia River down in the Oregon and Washington area. So the impact of predation on survival has remained poorly understood. So it's really great to see some of these, PDAT tags that are able to actually conclusively link predation to survival. But one of the big things that's been hypothesized is that it's non-native predation or predation in general, which may result in some of these much, much lower survival smolt outmigration numbers relative to the northern rivers. The Sacramento San Joaquin is one of the most highly invasive ecosystems in the world, as many of you probably know. So in addition to native predators like the Sacramento pike minnow, which can take a large toll on these outmigrating small fish, we also have a whole slew of non-native piscivorous, which could present a substantial risk to outmigrating Chinook. So that includes the striped bass, which you see here in the figure on the right, along with largemouth bass, channel catfish, white catfish that get more abundant as you go further downstream. So just a brief description, one of the great things and unique things about this system, and this has been pointed out in several other talks, is the Central Valley, the Sacramento San Joaquin, is an amazing area that's home to one of the most diverse suites historically of Chinook salmon. And this is embodied by 4 separate ecotypes, which are all in decline relative to their historic abundance. The fall run, which are currently the most abundant, late fall, which have winter migration timing, which may give them a little relief from some of these environmental sensitivities like temperature, winter run, which historically spawned above what are now dams, their natal habitat has been cut off, and the spring runs, which are just a shadow of the former abundance, but historically were one of the most abundant. And the winter and spring are currently listed as endangered and threatened, respectively, under the Endangered Species Act. So other than, just different spatial aspects of some of these fish, one of the things that really sets apart these different ecotypes is their migration timing and how they're moving throughout the system. So that's not only when the adults are returning back to spawn, but the amount of time the juveniles are rearing in the river and the timing of when these smolts are out migrating to the ocean. And you can see that to the right here, a basic illustration of how each of these ecotypes has specific differences in timing of when they're rearing in the river and when they're going to the ocean, which basically brings us to the research objectives. Based on these differences in timing, differences in size, differences in spatial aspects, the conditions experienced by each of these ecotypes of outmigrating smolt can differentially affect mortality and or predation pressure, which could preclude a one-size-fits-all management approach. We can't just tack a number and say, flow at this level is going to promote survival. This may be highly, context dependent on these different ecotypes. So our objective is to provide managers with information which can help address different sources of mortality at different times of the year for each of these different Chinook ecotypes. So again, this is a massive project that goes far back before me and is really still a very active area of research throughout the Central Valley. So for this particular analysis, we're drawing on 10 years of data from 2012 to 2021. And this not only allows us to really drastically increase our sample size relative to previous studies. Here we're drawing on about 10,000 tagged fish representing all 4 ecotypes, but it also allows us within our models to examine a much larger suite of environmental conditions to be able to draw inference on where some of these survival pinch points may be found for the different ecotypes. And where we have 200 acoustic receiver locations that I'll explain a little more in the subsequent slides. So as was touched on in the previous talk, what we're using here is acoustic telemetry data. So we basically have tags that have been implanted in these fish. These are juvenile salmon acoustic telemetry or JSAT tags, but a small tag is surgically implanted into these smolts, primarily at the hatcheries. These fish are allowed to recuperate and then released into the river, as you see on the left of this conceptual diagram. On the right side, we have acoustic listening stations or receivers that are put in fixed locations throughout the river. As the fish swims by, roughly within maybe 100 meters or so, that detection of a unique individual, time and location is associated, which allows us to track the movement of individuals throughout time. So of those hundreds of different acoustic stations that have been deployed over the last decade plus, what we've been able to do is basically reduce those to common pinch points and kind of areas that were always deployed over the years for the most part, and basically break this down in the upper Sacramento River into 14 distinct river segments that we can then assess survival as these smolts are moving downstream. So these different areas allow us to examine survival, detection probability, things along those lines at a spatially explicit kind of a level. So the long and short of what we're doing is basically to look at predation and to look at survival. We're using Cormack, Jolly, Sieber, Mark recapture models, which have been widely used to understand the survival and the detection of animals that are migrating unidirectionally through a river system. And since these smolts have to go to the ocean to complete their life cycles, that really allows us to understand that survival going downstream. If they stay, that they can't complete the life cycle, so we know that they have to continue to move downstream. So we're implementing this in a Bayesian framework, and we've adapted this code to the Nimble language. This is something that we were previously employing in JAGS, which has been used a lot in the past, and I'd be happy to talk with folks about this if they want some more information, but Nimble is incredibly powerful and incredibly efficient as far as what I've found. So models that would take maybe 3 weeks to run in JAGS are reduced to a several-day runtime in Nimble. So this has really increased our ability to utilize these techniques. So the models estimate 2 key parameters: apparent survival and detection probability. But the novel thing of what we're doing here is actually within that CJS framework, we're modeling survival as a function of predation risk using the mean free path length model, which was developed by Anderson et al. To understand smolt survival in the Columbia River up north. And just as kind of a crash course on the XT model or the mean free path model, basically it's generically referred to as the XT model because there's really only 2 key pieces of information that feed the model to look at predation. One, which is going to be the distance, X, and T, which is going to be the time of migration. So the way that we kind of think about this is as prey are moving downstream, survival is mainly an aspect of predator encounters as you move from one basically predator to another. Since the movement and the unidirectional movement of the smolts basically outweighs potential foraging of the predators, a lot of this is kind of looked at of moving downstream from one predator encounter to another. So there's 2 key parameters that are estimated by the XT model. One is lambda, or the mean free path length, which is the mean distance traveled, which is estimated before a lethal predator encounter. The other is the random encounter velocity, or omega, which basically is the time aspect of the model, or the mean encounter speed between the predators and the prey. To make this a little more simple too, basically The lambda, as that parameter increases and basically the omega decreases, it really suggests a true gauntlet-like situation where it's the movement of the prey and the distance of the migration, which is more important than the time. As that omega increases, basically that could suggest, one, that travel time could have a much larger impact on survival, or potentially search foraging speed of predators could have a large impact on what we're seeing. So within this mean free path length model, we're using ecotype and environmental covariates to inform these lambda and omega parameters. So specifically, we're looking for each release group at the smolt size. We're also looking at the mean annual flow across the duration that these release groups are migrating. And then we worked with NOAA to be able to model the actual specific reach-specific temperature and flow for each of these individuals migrating downstream. Again, we have time-specific detections for each of these smolt through acoustic telemetry. So using the raft model, we can basically tack on for each of these river segments an estimated temperature and an estimated flow corresponding to survival in that reach at that given point in time. So we've also included interaction effects in the models that allows us to examine how these relationships may change as we look from one ecotype or one release group to another. So moving into some of our results, what we find is that there's wide variation in the predation parameters, the lambdas and omegas, among river reaches, which, as we hypothesized, does suggest a non-uniform risk of predation among ecotypes and along the migratory corridor. But what I'm going to get into here in a few slides is basically some of the technicality of some of these results. So there's a lot going on here. And again, what we see is wide variation among reaches and among ecotypes as we're moving downriver. But rather than focus on these specific values, one thing that I want to kind of illustrate here, as you look at the left, the blue pane of this illustration, the lambda, or the distance traveled before an estimated lethal predator encounter, what we see is quite a bit of variation as you move from one river reach to another. The right-hand side, not only do we see these are very low omega values, but we also see a very, very small amount of variability, which suggests that, at least in the upper Sacramento River, this distance traveled is a much more important thing than travel time, and suggesting kind of gauntlet dynamics rather than active foraging of predators in that upper stretch of the river. So again, we've got a lot of variation here, but I'm going to kind of emphasize a couple of important points. So these are the environmental interaction parameters that go into the lambdas and the omegas. Basically, the distance traveled before a lethal predator encounter or travel time and how the environment may potentially affect that. What we see here, one, is that reach-specific temperature, that specific temperature and flow when these fish are migrating through seems to be the most sensitive as far as estimating or basically inferring predation relative to mean annual flow, which here doesn't seem to have as much of an effect. But again, less so the numbers that we're seeing here. What I want to illustrate is for that omega parameter, or basically the travel time aspect of the mean free path length model, we again see with our Bayesian mean estimates and credible intervals, we see a lot of overlap of 0. So these environmental parameters seem to have less of an influence on the actual time aspect than the travel distance as far as predation. And I'm going to talk about that a little bit more in a second here. So actually, I'm going to mention that right now. So out of the last conversation I've had with some folks about potentially these lack of sensitivity of the omega. A lot of questions have come up as, well, do you then need to include things like environmental covariates on the omega on the model? Or if you have lack of sensitivity and you seem to be getting consistent low estimates among river reaches, could this be modeled as a fixed parameter? So over the last few months, we've actually been performing model selection techniques where we've excluded some of these environmental covariates, not only from omega, but we've also modeled a fixed omega rather than a spatially variable omega. And while it seems like we're not getting a ton of information from this travel time, the best models are actually our full models. So while we do see a lot of overlap and lack of sensitivity, what the models are telling us is that including some of these covariates on the time aspect is still giving us some information and worth including in the model. So diving a little bit deeper into these specific environmental interactions, when we look at kind of a more generic course level across all fish, what we see is that this time reach varying flow is the most sensitive parameter for survival and for determining travel time or travel distance before a lethal predator encounter. So you can see in the upper left here, we have a very kind of strong positive relationship with flow. As flow increases, survival increases. And we have a little bit of an influence with temperature, negative relationship here as temperature basically decreases, survival increases, which is an intuitive thing with salmonids, which are a cold water obligate, and a little bit less or more of a dampened relationship with the mean annual flow and the fork length. But again, this is somewhat generic. So the whole point here is to be able to differentiate some of the specific environmental sensitivities of each ecotype, which may be very different from one time to the next. If you're migrating in the middle of the winter relative to the spring, if you're a larger fish, if you're a smaller fish. So now we're going to look a little bit more into the weeds of some of these environmental relationships. And again, here it's difficult to parse out. We see positive relationship with flow, negative relationship with temperature, muted relationship with mean annual flow and fork length. But this picture really does change when we start to look at some of these specific interactions, which I think is really cool. So what we've got here is basically your y-axis. We've got it divvied up by our different runs, the fall run, late fall, winter, spring. And then on the X, we see different columns of the mean annual flow, time reach varying flow, reach varying temperature. So I think a few interesting things to point out here is one, even though we did see at a very overall kind of a generic level, not a whole lot of influence to the mean annual flow, what we see here on the upper left is that we actually do have a pretty strong positive relationship with the fall run as far as survival or predation vulnerability based on the mean annual flow. And this really is something that I think is quite interesting. These fall run fish are going downstream in kind of coincidence of these upstream migrations of the striped bass when they're coming into the system. So potentially these higher levels of flow, higher levels of turbidity may provide additional cover as these fall run fish are migrating, which may not be the case if you're migrating in January or February when the stripers are still downstream. That could explain some of these relationships. Another thing I think that's really cool here is that we see temperature in a very variable kind of a relationship. Again, with the fall run, negative relationship. As temperature increases, survival decreases. Late fall, we actually see a positive relationship, which basically kind of counterintuitively suggests that temperature decreases may also decrease survival. Although through some of these years, 2019, 2021, we had very cold temperatures in the upper river. So maybe a very slight increase in temperature could favor metabolism and migration efficiency. So in conclusion, these results are suggesting a gauntlet-like predator-prey dynamic for Chinook in the upper Sacramento River, where distance traveled has a greater impact on survival than the travel time. Time-varying reach-specific flow has the largest single effect. We know that flow is a master variable, and in a highly regulated system, this makes a whole lot of sense. And also, we see differential effects of model covariates among ecotypes, which illustrates varying predation risk, which is likely tied to this migration timing. So this can be important to management because it can highlight portions of the river which correspond to low survival and may benefit most from management actions. We could also use some of these ecotype-specific parameters to inform life cycle models to understand different water management strategies and future conditions on survival. And we could also use these models to explore how management actions, e.g., flow release strategies, could affect survival. So we're currently in the process of extending these further downstream to the central or to the delta. So there's more to come on this as the fish go into some of the lower areas of the river where those lambda omega relationships may change quite a bit. So with that, I'd like to thank the many people who have participated on this, and I'd be happy to take any questions if we have time.
So unfortunately, we don't have time for questions, but please try to hunt down Jordan in this next break in about 40 minutes. Thank you, Jordan. All right. Next up, we have Matt Peterson with FISHBIO. If you want to use a pointer. Oh, okay. Then when you're done with it, just so you go to the next slide, click off. Okay, sounds good.
All right, thank you, everyone. Good morning. My name is Matt Peterson. I'm a biologist with FISHBIO. I work out of our Chico office, but today I'm going to head south a little bit and talk about some work that we've done on the Stanislaus River and really kind of do a deep dive. The previous talks were really good about kind of looking at big picture relationships between predation and survival of salmonids. Today we'll take a kind of a deep dive, and this will be kind of a high level culmination of our work on the Stanislaus River, which was begun in 2017. Well, I thank my co-authors, which were listed on the previous slide, our funding sources, and lots of folks that helped out on this project. Like I said, we began in 2017. We did roughly 5.5 seasons of field work. So a lot of folks helped out. And I should mention that this work was prompted by the WIN Act that was passed in late 2016, which required the Oakdale and South San Joaquin irrigation districts to work collaboratively with NOAA Fisheries, and in addition with CDFW to implement the study, or this suite of studies, I should say. So today, I'll give you a quick background on the Stanislaus River and some of the project objectives, talk about the methods, what we learned, and today I'll really focus on the predation impact from piscivorous fishes. That's the main goal of today's talk. Talk about some of the next steps that we're working on, and obviously taking questions if we have time. So Jordan gave a great background on, kind of the large scale impacts of human activity in the Central Valley. Don't really need to rehash all those, but I'll just say that Stanislaus River is, a great example of lots of those impacts on anadromous salmonids. The Stanislaus River has benefited from a long-term salmonid monitoring program that really began in the mid-1990s. And this is focused, a lot of this work has been focused on this highlighted area here in orange. We know a lot about salmon population dynamics in this highlighted area. And so we have pretty decent estimates of adult abundances, juvenile abundances. And then there's 2 rotary screw traps, which have been run since the mid-1990s. And really, that data was used as the impetus for like this larger research program. We documented very poor survival between those 2 traps. Those traps are only located, or the distance between those 2 traps are roughly only about 25 river miles apart. And on average, across about 25 years, we'd lose up to 80% of our salmon in between those 2 traps. And before this study, there really wasn't much known about the factors why we were losing so many fish in that section. So beginning in 2018, when the field work really started on this project, we started the project using what's called a robust design sampling framework. And this allowed us to repeatedly sample our sites. We had 39 different sites, which I should have pointed out on the map. 39 different sites located from Oakdale down to the confluence of the Stanislaus River. Those sites were repeatedly visited each year for 5 years. And so some of those sites were sampled up to 48 different times, 48 different days. At each one of those sites, we basically performed a mark recapture study of both native and non-native predators, and we collected their diets. So those are the 2 big data inputs that go into this. The nice thing about the robust design sampling framework is because you can break the data down into different time periods, we're able to estimate the annual abundance of our native and non-native predators within each sampling period. So we really focused on the main outmigration period of salmon, which on the Stanislaus River is February to June. So how the field work was carried out, we used boat electrofishing exclusively to capture our predators. This is a quick shot of one of our cataract electrofishing vessels, which is sampling at one of our units at low flows. At higher flows, we had to use jet-powered vessels. We sampled February to June for 5 years, so really got to get some wide environmental variation on the river. We sampled roughly from 250 CFS all the way up to 5,000 CFS. Basically in 2019 and 2023, we had some really wet conditions, so we got to experience some really wet years. And then from 2020 to 2022, we had much drier conditions, and so had much lower flows, warmer water temperatures, and such. I should note that I'm blanking out right now. Excuse me. I should note that water temperatures during our sampling ranged from about 9.5 C up to 18 C. We weren't able to sample above 18 C due to regulation, permitting restrictions with boat electrofishing. So that's kind of a big state limitation. So in certain years, we weren't able to sample past about mid-May because temperatures would exceed that 18-degree mark. So at each site, we collected our predatory fish, whether that be native or non-native. I put in a couple shots of small Mycropterus here. For the purposes of this talk, we're really just focused on the 3 main predators that we observed, 3 main predators on salmon. So Mycropterus, and I'm pulling smallmouth bass, spotted bass, largemouth bass, and red-eye bass all into one. Striped bass and Sacramento pike minnow were the main 3 players. We did observe 1 hardhead consuming a salmon, but that was one of about 250 that we sampled. For every individual predator that was captured, we pit tag them, measure them, pump their stomachs using gastric lavage, and release them back into the site. And then for each diet item, we would either use visual identification techniques or genetic identification techniques. And we had to use the genetic ID for items that were heavily digested. So across the entire 5 years, as I mentioned, we sampled a lot out on the boats, 300 hours of electrofishing, captured roughly 15,000 fish in total, 10,000 of our target predators, and pumped roughly 6,000 stomachs. And then all told, the diet items from those 6,000 stomachs, I believe we identified on the order of 40,000 individual diet items. So there's a lot going on here. I will try to make this as simple as possible. So this is our very complex model to estimate the predation impact. And I just want to stress that there's 4 main ingredients that we used, really 5. We used a gastric evacuation model for each one of our main predators that we saw consuming salmon. So we had one for black bass or Mycropterus, one for Sacramento pike minnow, and one for striped bass. We obviously had their diets. And again, just going to focus on Chinook today. I would refer you to a paper we recently published on kind of the larger, broader diet composition that we characterized in the first couple of years of the study. So we had diets from the predators. We used, because we had these repeated counts of our predators at each site, we were able to use Dale-Madsen in-mixture model, which is a dynamic in-mixture model, which accounts for detection both during the closed periods and survival in between the closed periods. And so we're able to estimate the annual abundance of predators in the Stanislaus River annually, and then also get uncertainty with those associated with those estimates. I know these graphs are small. Highlight is that the predator populations did change substantially over the course of the study. Had an increase. We observed an increase in Mycropterus over the study period. Observed an increase in both Sacramento pike minnow and concurrently hardhead, which are not shown here. over the study, and actually a decrease in striped bass in the river. So that's kind of the third main ingredient. Fourth main ingredient is a spatially explicit water prediction model that we developed using temperature logger data that was collected throughout this reach, but at a lot of the sites we didn't actually measure, or we didn't have daily water temperature at each one of the sites we actually sampled. So developed a model to predict what the water temperature would be at each one of those 39 different sites on each day. That was fed into the gastric evacuation model. And then I mentioned, it's all well and good to estimate how many Chinook could be consumed by predators, but without putting it into context with the availability of the prey that we're interested in, it's really meaningless, right? So because we had concurrent rotary screw trap monitoring, we were able to estimate abundance of juvenile Chinook salmon moving into the migratory corridor on a weekly basis, and that allowed us to get at the actual predation impact. That's our denominator in the simple calculation here. And then I give a shout out to our agency partners, both CDFW and NOAA Fisheries, who during an interim review of our program, I believe it's back in 2020 or 2021, they really stressed that we needed to not only get the point estimate of what the predation impact would be, but because we have such a complicated model, we also needed to carry forward error in each step. And so how we did this is that we tried to either estimate the error directly on each parameter going into this big model, or assign it some kind of arbitrary error, and then used a resampling procedure and a simulation process to basically redraw samples every time, usually with replacement, so that we could assess, so we could first assess the uncertainty, and then coupled with that, because there are a lot of moving parts in this model, we also wanted to see how the different input variables, how those might affect the ultimate estimates. And we did that using a one-way sensitivity analysis. And so really, again, I'm going to stress these four main components. I'll really highlight those today, but we had 14 other ones that also were fed into this model. So very complicated. For each one of those 18, we varied those in different ways to assess sampling variation, and also any methodological issues that we may have introduced into how we did things, to see how those would affect our predation impact estimates. We had 5 years of data, made 72 input variables, which were varied one way or the other, 250 simulations. So in the end, we did 90,000 simulations to assess the error and how this might propagate forward. At first, I just want to touch on what the point estimates along with the error estimates show. Really, we saw variable impacts from Micropterus, fairly much more consistent impacts from striped bass and Sacramento pike minnow. And when you add these up across those three main target predators that we examined, we estimated that during our predator sampling events from 2019 to 2023, these piscivorous fishes could consume between 20 and 60% of the salmon moving into this corridor. And then on to the sensitivity analyses. So I'll just first give everyone a quick rundown on how this is laid out. So the gray diamond is the point estimate from the previous slide. This is the total, so this is combined across all the three main predators. This is data just from 2019, and I'm just highlighting 2019 because it's, it was a wet year. It's pretty interesting that we had roughly 21% loss in this reach. And of the four main ingredients that were fed in, water temperature came out to be the most important. And I should have mentioned that we varied water temperature in a somewhat crude way, maybe not too realistic. We just used a blanket assumption of we added 2 degrees to kind of mimic a potential climate change scenario. Well, how would this predation impact increase in a warmer climate? And then we also decreased it by this blanket assumption every day at every site for every fish that was fed into this, in the event that you could potentially manage the water system in the Stanislaus River a little bit differently to try to see if you could decrease this predation impact. And so because we use such kind of a, I would say kind of a crude, a crude adjustment of water temperature definitely came out to be most important, followed by Chinook abundance. So if we're off in our estimate of Chinook abundance at the trap, at the top of the reach, if our true estimate was here and we actually estimated this much, the predation impact would be slightly lower. So that was the second most important overall. Obviously, the Chinook prevalence in the diet, if we messed up on that estimate, going into this larger model, that would affect it, as did striped bass and Micropterus estimates. And I should say, just highlight 2019 because it's a wet year, but the results were pretty comparable across all years. And to save everyone, because I had such a complicated model, I'll leave you with just one main takeaway for today. Across these 5 years on the Stanislaus River in the lower between Oakdale and the confluence, we estimated between 20 and 60% loss of juvenile Chinook salmon in the river. Currently, we're working through the final stages of revisions on our report. We're continuing to try to publish and share findings as we can, and really excited to, we're in the final steps of almost submitting a paper looking at the spatial patterns of consumption across this reach, which can really allow us to do a deeper dive on how the underlying prey availability, how habitat, and how our environmental conditions might affect the probability of consumption of salmon at these different locations. And with that, hopefully we have a couple minutes. I'll take any questions.
So we have about 2 minutes for questions. Got one online?
Yeah, we've got one from Trevor. Given that bass abundance had the lowest impact on your input variables, how do you view the relative value of predator removal interventions versus water temperature, et cetera?
Currently, it would appear based on our model that water temperature would perhaps produce a more robust effect on lowering that predation impact. But as I mentioned, the way we manipulated water temperature within this model was relatively crude. And I would say that we probably would need to actually run some models to estimate what you could feasibly get in terms of water temperature reduction from Goodwin Dam and New Maloney's Dam to really compare those two options. And that is definitely a next step that we'll hopefully be undertaking.
I think we're out of time for that. Thank you, Matt. If you have any questions, come find them in the break. Yes. All right. So next we have Wim Kimmerer with San Francisco State University.
Thank you. All right, now for something completely different, as you probably expected. Not a word about salmon. So we have 4 introduced species of copepod whose distributions overlap, and that's what I'm going to talk about, how they accommodate each other and the system. So I'm going to introduce the players first, the 4 players, and Pitamacorbula amurensis, that you know all about. Talk about potential interactions and then see which potential interactions might actually work and why competition is unlikely. So, so the players are Eurytemera carolee. Now I'm calling this introduced because even though we used to think it was a native, it's not. It was introduced probably in 1879 when the striped bass were brought in from the East Coast. Pseudodactylus forbesi, introduced in around 1988, and then Acarciella sinensis and its main prey, Limnoithona tetraspina and sinensis. And so Acarciella, unlike the other three are particle feeders. Acarciella sinensis is an obligate predator. It doesn't eat phytoplankton. It eats other copepods. And a clam. Okay, so the clam's going to play a key role here, even though I'm mostly talking about the little bugs. So a little background for the copepods. I use a salinity-based frame of reference instead of geographic. And the reason is that that's the frame of reference these bugs live in. And so if you track them in their own frame of reference, you can learn things that you would not learn by looking at them in a geographic frame when movement of the water mass would confound things. They're undergoing a severe food shortage, particularly the particle feeding, the 3 particle feeding copepods. And I'll show you a little bit about that. No surprise. What about predation? I mentioned that the Acarciella sinensis is a predator. And so that's the main predator I'm going to be looking at, but there's obviously a lot of other predators I'm not going to talk much about. And then competition. And there's a question mark there because actually I don't think it happens. But if it does happen, among these particle feeding copepods in particular, if it does happen, we would not be able to detect it unambiguously. So this is the abundance history of the 4 species. And I'll just kind of quickly go through them in order. So Eurytemera carolee was here when the zooplankton monitoring program started in 1972. And you can see their abundance kind of bounced around and without a seasonal pattern to speak of. And then abruptly, when Pitamacorbula arrived, their abundance declined about tenfold, and they switched to an annual cycle where they're abundant in the summer or in the spring and rather rare through the summer and fall. And that's about the time also that Pseudodactylus forbesi showed up, about a year after the clams showed up. And they also have an annual cycle, but they're most abundant in the summer. So I'll talk about this a little later. You know, might look like the result of a competitive interaction because they're both particle feeders, but it doesn't seem to be the case. Then we have Acarciella and Limnoithona, which were Limnoithona tetraspina, which were both introduced in about 93 and both also became abundant. And they also have annual cycles, and they're also most abundant in the summer. Pseudodactylus forbesi, Acarciella sinensis, and Limnoithona tetraspina are all tropical to subtropical species. So in the winter, they're kind of gone. So what causes these interannual cycles? Well, here's a really important component of this system. We know it's important, but some people seem to forget it. Clam grazing is an enormous source of mortality. So this is clam grazing on grazing rate of the clam assemblage on particles in phytoplankton in the and microzooplankton in the low salinity zone, 0.5 to 5 salinity. And there's also grazing on the nuclei. We've shown that at about 10 to 20% of the rate of the grazing rate on particles. So that imposes a mortality that's about 10 to 20% of what this is. But notice that this is on a log scale. So it goes as high as 1 per day, which is way above the replacement rate of the phytoplankton and the microzooplankton. So So that has a huge impact, and you can see it in this graph. Chlorophyll concentrations during March and April, May, June, July, August. And you can see how over time, there's this, a lot of variability, but basically the chlorophyll level, which is a measure of phytoplankton abundance, biomass, it declined sharply when the clams showed up, especially in the summer. And these horizontal white lines are the band of sort of food limitation that I'll show you later of the zooplankton. So that's IEP, EMP survey data in the green dots and the orange line, and then the other one is the USGS survey data. So food limitation. So this is for Pseudodatmus. And so this is a concentration of chlorophyll as a measure of phytoplankton biomass, and then their growth rate. And you can see most of the data points are down here in the low end. This is the 1 in 10 micrograms chlorophyll per liter lines that I showed before. And so you can see they're severely food limited in their growth rate most of the time. And the same is true of their egg production rate. So that means that their population dynamics is severely affected. So this graph, I have to introduce you to it first. It shows the abundance of each species, of these 4 species, as a heat map. And the y-axis is salinity. It's actually on a log scale, and the x-axis is month. And the brighter colors show you the higher abundance of these species. And so going from the upper left, we've got Eurytemera, which has it's most abundant in the spring, and then it virtually disappears in the summertime. Big hole there. And it's most abundant in the low salinity zone. Pseudodatmus is most abundant in freshwater. Earlier in the season, it's very abundant in the low salinity zone, and it shows up around May or so, roughly the time when Eurytemera disappears. And then Limnoithona is most abundant, higher salinity than the other two, and kind of highly abundant throughout the summer, well, spring through fall. And Acarciella sinensis, pretty similar, a little bit less in the springtime. So what are the consequences of that? What can we infer from this? So the first thing is, you look at a graph like this and you say, well, what's going on here? Why is Eurytemera gone in the summertime? And you might think it's some effect of competition between the two copepods, the two larger particle feeding copepods. And that's sort of a logical assumption. But actually in the literature on competition in zooplankton, there's almost none reported. And most of what you see when people say there's competition between zooplankton, they're either inferring competition from overlap in species in distribution, in time and space, and in diet, or lack of overlap. People use either one of those to infer competition. In the first case, they must be competing because they're in the same place eating the same stuff, or they're not in the same place because this species is out competed in this area where the other one is. Can't have it both ways, right? And there's not enough information from that, from those things I just described, like these graphs, to infer competition. You actually have to look at what they're eating, and you have to look at whether one affects the food supply of the other. And that's extremely difficult. And as far as I know, it's only really been done in lakes with no fish in them. And the reason for that is predation is such an overwhelming process determining population dynamics that it really pretty much wipes out any kind of competition that's going on or makes it invisible. So I'm going to show you some data that suggests that this interaction is not happening. It's also possible that Limnoithona is competing with Eurytemera, but that's also very unlikely. There's also a possibility that the predation by Carciella on Pseudodapnus and on Limnoithona is important in regulating their position. And I'll show you that that's, in the case of Pseudodapnus, that's true. So Limnoithona, it's like, I forgot to mention, is about half the size of the other, half the length, and so about an eighth the biomass and per individual. And that's like Sinocalenus's bread. That's their main diet is Limnoithona. So Limnoithona is kind of like a background abundant food source that keeps Sinocalenus abundant too, and available to prey on Pseudodapnus. So here's some predation data from a paper we wrote in 2016 or published in 2016. So these are just showing predation rate of a Carciella feeding on Limnoithona life stages. Adults, nothing going on. Copepodites, not much going on. The Nauplius larvae just get hammered. So these clearance rates, which are sort of like well, they're mortality rates. So mortality rate of 20% per day on the Nauplii of Limnoithona, and at about 60% per day on Pseudodapnus. So they really don't stand a chance where there's overlap. And I can show you the time frame of that. So here is, this is chlorophyll, Pseudodapnus abundance. It's actually frequency of samples greater than 100 per cubic meter. And the hot numbers are, the hot regions are the high frequencies, 100%. And you see this abrupt drop that happened in 1993, and guess who showed up then? Carciella. So, we know that a Carciella showed up then. We know that a Carciella preys on the Nauplii of Nauplius larvae of Pseudodapnus and other copepods. So what about competition between Eurytemera and Pseudodapnus? Well, I don't think it's important for several reasons. This is some work that Amy Wong did in my lab, using DNA to identify the prey of various of the two species, the two larger calanoid particle feeding copepods. So this is what was in the water, different taxa, phytoplankton and microzooplankton, and this is what was in the copepods. And there's almost no overlap. Now, this is so startling. I mean, all our jaws dropped when we saw this, when Amy first presented this to our lab. And it was so startling and so unexpected and so complete that we actually did the experiment over again, and the samples are just now getting in the queue for sequencing. So we'll see what happens. I'm nervous about it either way. Either because I can't explain it or because I can't explain it. I can't explain the difference in results, or I can't explain their concurrence. And then finally, there's another way of getting at this idea of competition, which is where Pseudodapnus is abundant, Eurytemera should be declining rapidly. So I took all the data since the time when they were both present in the system, and I looked at the spring decline of Eurytemera, which is falling off a cliff, and so each spring they decline at a certain rate, and this is the rate of change here. So the median is something like 8% per day, and I related it to the clearance rate of particles by Pseudodapnus weighted by the abundance of Eurytemera, right? So we're getting at the abundance of Pseudodapnus where Eurytemera is abundant, because where Eurytemera is not abundant, we don't care. And then this is the relationship. This is probably the most scattered diagram I've ever seen in my life. And there's obviously no relationship at all there. So in other words, consumption of particles by Pseudodapnus where they overlap, even if you don't believe that diet stuff I just showed you, consumption of particles by pseudodaphnis is not an explanation for the rapid decline of Eurythemera when they fall off their cliff. So, key lessons. Well, the first one you obviously know because we've been talking about it for the last couple of days. But this is a particularly complex estuary partly because of the huge amount of data we got. This is probably, well, I'd say it's currently undergoing the most study of any estuarine river system. I just wish we were paying more attention to the saltier parts of the estuary because it is a whole system and we're neglecting the salty parts. Then competition between species of copepod is rarely detectable, and if at all, where there are fish around. And there are definitely fish around because that's a major source of losses to these populations. And then predation is, in general, I think, by far the stronger interaction than competition. For competition, you have to have, there's 3 steps involved, right? I'm eating this, you're eating this, and we're affecting the food, or you're affecting the food, and I'm losing out. And whereas, I get eaten, well, that's it. So thank you for your attention, and maybe I have time for questions, I don't know. That says, arigato gozaimasu. I'm told.
Yeah, you have time for about 1 or 2 questions. Got a hand in the back.
I'm wondering what the sort of process is for, I guess, analyzing the stomach contents or the feeding habits of something as small as a copepod.
We do not dissect them. So the only graph I showed of the actual stomach contents is based on DNA. So we just smoosh them up and process them for DNA. People have done that, but it's really, you can only look at hard parts. It's impossible to really do anything useful. The other option would be stable isotopes, but that's too crude and coarse-grained. So the only way to do it is really with DNA. But with those results, we're kind of questioning, is there some part of the process that bias our results? And we'll find out, I hope, in about 2 weeks. Okay.
All right, thank you. All right, next up, we have Francisco Villero Leyva from UC Davis.
Hello. So today, I'm gonna move a little bit of system. I'm gonna go way more upstream to ecosystem just downstream reservoirs, focusing mainly on the Shasta-Keswick complex and all the work we have been doing there for the last 4 years. So as we all know, dam has disrupted ecosystem processes through many different pathways. So they have altered natural hydrographs in this downstream ecosystem. They have also disrupted the historical temperature regime in this ecosystem, both for cold water native species or even warm water native species. But they also have changed the sediment transport, and that also changed the complexity of habitats downstream, the amount of floodplain, the amount of channel habitat, how that is connected to the main channel. And also they have disconnected many species from the historical habitats. They have disrupted this longitudinal connectivity that used to happen in all the Central Valley. So we have seen how all these effects have produced declines in native species in many different systems. Here we have winter run Chinook in the Sacramento. So we have seen how those declines have happened throughout the years due to the impacts of dams. But we have to acknowledge that dams provide a lot of societal benefits, and they are providing for agriculture, urban water use, flood protection, and hydropower production. So here we have kind of a snippet from the hydropower vision report, where it's indicated that hydropower is a renewable source, so it's expected to increase in future years as we kind of go off non-renewable energies in many different areas. So we need to start thinking how we can operate these reservoir dams in order to provide some of these ecosystem processes that we have lost throughout the years. So we have started thinking about this in the last decade. So for example, we have the California Environmental Functional Flows Framework. So where we want to mimic certain important parts of the historical hydrograph to produce this kind of processes that we expect native species to have developed and evolved to take advantage of. We can also produce, try to manage for historical thermal regimes in these downstream habitats. But for this, we need to have some kind of infrastructure that allow us to extract water at different depths for different temperatures. But also when we think about these, having this infrastructure in these reservoirs, and we are selecting where we are putting water out in the system, we actually also are going to influence, which is the water quality of the rest of the water that is being released. So for example, here we have some, the example of Shasta TCD where we can see how water has been extracted from different areas. So for example, a mix of epilemion and hypolemion waters at a certain time of the year, epolimnetic waters another time, or epilimnetic waters at different times. And these water will have different water quality constituents within them. That is going to affect what is being seen or what is being exported to the downstream ecosystems. So the question there is then, can we explore that differences in water quality as a potential for providing flexibility to management in these very limited systems? So kind of this question arose from a pilot study my co-authors did from 2018 and 2019, where we sample nutrients and zooplankton, phytoplankton in the Sacramento River downstream Keswick Dam. So here we have the conditions on Shasta, temperatures, and each one of those values representing which TCD gates were open, and the vertical ones when we were sampling in the system. And we observed in the samples we took in the Sacramento River that nutrients that exports and nutrient concentrations in the Sacramento were highly correlated to deeper releases in the reservoir. While we also observed high concentrations in food web resources, both phytoplankton and zooplankton in the Sacramento River during this time. And they were more associated, mainly zooplankton, with the shallower releases. So during this whole study, we measured an estimated 95 metric tons of zooplankton being exported to the downstream ecosystems. And that's a lot of food. That's approximately 10,000 bags of happy chow that they were exported as carbon in an ecosystem, in that downstream ecosystem. So we kind of from that initial analysis of just sample taken in the Sacramento River, we kind of got this conceptual kind of picture of it, how lower releases are producing this more kind of nutrient subsidy, while the higher releases during productivity periods in the reservoir are producing this kind of more food web subsidies to the downstream ecosystems. So that kind of gave us that question is can we provide additional flexibility to management in the system, taking into account these water quality conditions? So this is how our current project started. Is we only sampled that time in the Sacramento River. What is happening in the system? What is happening in Shasta? How much is exported from Shasta to Keswick? How that moves through Keswick? So that's kind of our main question. So which are the productivity dynamics in Shasta? Are they consistent? Are they just like a peak and then nothing happens there? Or how is the productivity? Can we take advantage of that throughout the year? How much is exported to Keswick with the operation of the TCD, how it was operated for the different depths, what we see in Keswick, what we see it routed through Keswick and arriving to the Sacramento River. And how much of these resources actually arrive to the Sacramento River, and then how are they used by the ecosystem? Are they directly used by the fish? Are they kickstarting productivity in an ecosystem and then the fish are taking advantage of that? So there is, all this question is what our work is now focusing in this area. So I'm gonna present like a couple of like general results. For example, here we have nitrate concentrations from left to right in these bars. We have Shasta Reservoir through Keswick until like the red bar that is the Sacramento River. And this, for example, I made a snippet from like October to January that we have lower releases in the reservoir under like the metalemia and some part of the hypolemia later in the year. And we can see how concentrations that we observe in Shasta are mostly exported through Keswick and arrive into the Sacramento River. So that nutrients that have been accumulating in Shasta while it was stratified during the summer, coming from the inflows to the reservoir, they're actually being exported to the Sacramento River later, like in fall, winter. Then we also observe, which is the secondary productivity in the reservoir, which is the zooplankton densities we observe. So here we have for the different years we sample. The blue bar is copepods, the green one is cladocerans, and we observe high productivity that happened at the onset of stratification in the reservoir, and it's something that we should expect in lotic environments. At this time of the year in our temperate climate. But we can see how that peaks in productivity actually are moving throughout the system. So that productivity happening in Shasta is being exported and moved down to the Sacramento River. But we actually observe a difference in the concentrations we sample in the Sacramento River between all the different years we were there. So here in the left, we have during our pilot study in 2018, and then in the right, we have 2021 and 2022 that we were sampling out there. And we can observe how in 2018 and 2022, we have much higher concentration of zooplankton that we were able to observe in 2021. And so then we started thinking, why is this difference? We were sampling maybe when like we were offsetting timing, maybe there was not a peak in the reservoir when we were sampling in 2021, but yes, in 2022 and 2018, but we observed, I'm gonna go back one, that actually we observed high concentrations in the Sacramento, in Shasta Reservoir in 2020 and 2021 in the top there. So then we started looking at the configuration of the TCD at those times. So we can observe how both 2018 and 2022, we were releasing much shallower water from the reservoir than in 2021, that it was deeper. It was like over 40 feet in depth, that we were releasing to the Sacramento River. So then we started thinking maybe that's heavily impacting the amount of zooplankton we actually saw is being exported down to the Sacramento River. So then in 2022, we got on our boat, we put the biosonics on it, that is a high resolution echo sounder, and we started mapping zooplankton close to the TCD, and to see where the zooplankton was in the reservoir. It was only when we were sampling, it's more or less distributed, and it's in that kind of shallower part of the reservoir. And we actually saw that. So the clouds of points we see there is large body cladocerans that are the ones that were able to pick up with the echo sounder. We did some zooplankton throws also to kind of validate that what we were seeing there was zooplankton and not, I don't know, turbidity or other particles in there. And we actually saw correlation between what we saw in zooplankton and what was picked up by the echo sounder. And we can observe how most of the concentration of zooplankton was focused on that first 30 feet of the water column. So that kind of make us think or agreed with our hypothesis that those deeper releases in 2021 had a big impact of the amount of zooplankton we were able to see in the Sacramento River. So then, now the question is, how then is that used by the ecosystem? So that's kind of like our more current work right now. Here we have invertebrate density in the Sacramento River, Clear Creek, and Stanislaus, the CLC and STN respectively. And we could observe much greater concentrations, abundance of invertebrate in the Sacramento River than in the other two systems. So part of these nutrients and zooplankton move down to the system seems to to be producing high productivity of invertebrates in these areas that then might be able to have might be producing kind of a cascade of productivity. So we have invertebrates, and then that might be picked up by other higher order consumers and kind of like producing this high productivity in these downstream ecosystems. So in order to clarify that, we are doing some isotope sampling to see how is the structure of how is the food web structure in these areas. So we did some sampling in 2022 and 2023, but we're actually doing a more intense sampling this year. So that's us doing some sampling like just a week ago when we were sampling before the expected peak in productivity in the reservoir. We're gonna sample again during the peak. We're gonna sample after the peak. We're gonna sample again in October. So we can actually see if the food web changes when we are seeing those higher exports from Shasta, to have a much clearer picture of how those nutrients, that zooplankton is being uptaken by the ecosystem in those areas downstream the reservoir. And apart from that, we also have DO sensors downstream Shasta, where we can estimate GPP, gross primary productivity estimates in this area. So we can also try to think or try to observe is during the times we had greater nutrient exports from the reservoir, do we see an increase gross primary productivity in that area downstream the reservoir as well? And also that I didn't mention, we had so during our sampling for isotopes, we also take some fish out of the system, and some of them are also being processed for otoliths to see if we see changes in growth from those fish to other growth in otoliths we have in different areas around California, and observe if there is also a pattern of higher productivity and higher growth for fish in these downstream ecosystems. So the end product we want to have with all of this is then we have built a model for Shasta Reservoir, the C equal W2, where we are trying to model productivity and water quality in order to have a tool to kind of explore trade-offs of changing the TCD operation. So we can say to the model, what happens if at this time I open the higher gates, the change that we are going to expect in the Sacramento River in temperature, nutrients, zooplankton, and then start making some trade-off analysis of the exports of energy from the reservoir, also with temperatures downstream, how that affect the native population of fishes there. So we are building this tool with all the information from isotopes, otoliths, to understand how all the ecosystem is responding to those exports and explore those trade-offs in management to provide maybe additional flexibility in such a constrained system. So this is kind of like what we expect to see, like differences in exports with the model and understand how that affects the ecosystem. That's all I want to get through today. Acknowledgements also for everybody that helped us through all this research. Yeah, thank you very much. If you have any questions.
Francisco, we have time for a couple of questions. Thank you.
Thank you.
Thanks, everybody, for those excellent talks. We're going to take our first break. Please be back at 10:55. All right, everybody, we'll go ahead and get started with the next session. Nick, take it away. Nicholas?
All right, everybody, welcome back from the break. My name is Nick Rasmussen. I'll be the chair for our next session on management and policy. So we'll go ahead and get started with our first talk. So first up, we have Brian Maharsda from USVR with his talk entitled Decision Uncertainty and Research Prioritization for Delta Smelt Summer Fall X2 Action.
Thank you, Nick, and thank you all for coming. So today I'm going to talk about a decision analysis study that we did starting last year. And decision analysis is just a systematic approach to kind of consider all your options given a particular problem and to make a decision based on thinking of what the possible consequences are. All right, for a few of you who are not especially familiar with Delta Smelt, just a brief summary. The Delta Smelt is a small species that lives only for a year. It's only found in the San Francisco Bay Delta. It eats primarily zooplankton. It likes habitat that is cool, turbid, and anywhere between freshwater and low salinity. And the reason why we care so much about them is because they're listed under both the federal and state endangered species acts, which means that they affect water operations to some extent. Now, for the species, summer fall is generally considered a period of time that's a bottleneck because that's when the lowest flow is during the year for California. And outflow is one of the things that we can potentially control, as water projects. So given that, in the past, to increase the amount of habitat for smelt, outflow has been augmented in the fall, sometimes referred to as fall X2 action. But a recent study indicated that maybe summertime is better, period to kind of do this action. But it's not as simple as just kind of switching the action from fall to summer. There are a lot of things to consider, right? We live in a highly complex system and there's trade-offs to consider. Water that gets used for Delta Smelt could potentially be used for other things, including municipalities, ag, and even other species like salmonids. Okay, so the reason why we conducted this study is really to evaluate the roles of the values and uncertainty for decision-making surrounding this large-scale flow action. We also wanted to look at value of information. So this is, value of information is how much the expected outcome of a decision would be improved if we resolve uncertainty first before committing to a course of action. So in a sense, kind of gives you the value of like scientific research or study. And I just want to note, because we didn't really explicitly involve decision makers in the study, which would be difficult, this is sort of a decision aiding kind of study rather than decision making. Okay, first I want to kind of go over the mechanism of how we think flow, additional flow can benefit Delta Smelt, because it's not just purely correlational at this point. So let's say Delta Smelt are here in the summertime or fall. If we move the low salinity zone where Delta Smelt likes to be, we'll potentially move Delta Smelt downstream. This downstream area is typically cooler and has higher turbidity, which would lead to good things for Delta Smelt, like higher biomass and therefore survival, and therefore higher population growth rate. The other component is the food that they eat, the zooplankton or copepods, which we know can be transported downstream, which would give the fish better access to food, and therefore increasing their population potentially. But the other consequence of doing this action, right, we could potentially lower the water supply for other uses, anything that's not Delta Smelt. So those yellow things are the objectives. We also have uncertainties. So there are 3 uncertainties that we're gonna look at today. The first is kind of like how much or if this zooplankton really gets transported downstream, how Delta Smelt move with additional flow. And we're also going to think about bioenergetics and how these additional food or movement to a different region can benefit Delta Smelt in terms of its biology. Okay, so for this case study, we're considering multiple objectives. The first one is Delta Smelt. We kind of want to maximize the population growth for Delta Smelt using lambda, which is a term used commonly in population dynamic studies. Larger than 1 means the population is growing, less than 1 means it's declining, and 1 is self-sustaining but stable, but not really increasing or declining. For the second objective, it would be water. So we want to minimize the water supply costs, measured in 1,000 acre-feet for the action. And we're just considering the cost for a given action year in this case. All right, so let's go through the different options that we considered. So the first one is the status quo. So for the past, I think, 5-6 years or so, we are committed with the water projects have been committing to a fall flow action, maintaining X2 or the low salinity zone at 80 kilometers, which is the distance from Golden Gate Bridge there, focusing on September and October. And we also considered what if we kind of double down in the fall and increase even more flow in the fall. So this is sort of, but not exactly a proxy of a previous biological opinion in 2008 and 2009. We also looked at the summer action, which is kind of an approximate of a study of a hypothesized action that Leo Polanski and others at Fish and Wildlife Service kind of looked at in the recent paper. So moving X2 at 74 kilometers in July and August. And then what if we combine the summer and fall action, targeting 74 in the summer and 80 kilometers in the fall. Lastly, what if we just don't do any action? But I just want folks to keep in mind that the action only occurs during wet and above normal years as you interpret the results. And that across all these different alternatives or options, the baseline actions for Delta Smelt are present. So this includes the export management in the Delta for Delta Smelt and Susan Marsh salinity control gate action. Okay, to go over the methods real quickly. So the red is objectives. So first of all, we use calcium 3 to kind of predict how much water we would use for this action. It simulates about 100 years. We then get an output, including X2. From the X2, we have a couple of submodels that we use. First, we try to kind of predict how much zooplankton would be increased or changed really, because some might decrease based on the particular X2. We also look at how Delta Smelt move with X2, which get then plugged into this individual based model that was originally developed by Kenneth Rose and Wim Kimmerer. That was then adjusted further by Will Smith at US Fish and Wildlife Service. This individual based model, or IBMR, because it's in our programming language now, is simulating about 20-year time period, which would then give us lambda. Oops, wrong button. So yeah, and then there's the yellow here, which are the kind of the uncertainty that we're going to test. We're basically going to turn on and off these submodels. And on top of that, we have two versions of this IBM or individual based model. The first one is looking at monthly time step. It's closer to the original model with bioenergetic parameters borrowed from rainbow smelt, but adjusted. Whereas the second version works on a daily time step and it's based on our more recent paper, where the bioenergetic parameters were kind of adjusted further with more recent data. So the way we do this is that, you know, we turn on and off for the two submodel, and then we have the two versions. And the way it's framed in decision analysis is as different hypotheses or different beliefs of how the system works to represent uncertainty. So I'm just kind of laid out here to make it easier to understand. So we have two versions of IBM on the rows. On the column here, we have the full model where we allow Delta Smelt to move and food to move based on salinity. And what if we turn off one, which is the Delta Smelt movement, and just keep the food? What if we do the opposite, keep the movement on, turn off the food? And what if we believe flow just doesn't do anything? We turn off both of those. So in total, we have about 8 different model variants or 8 hypotheses. And then what we do is, what would you typically do is you have an expert elicitation to kind of figure out like, what are your relative beliefs of the different model types or hypotheses. So in this case, we just kind of relied on the set of co-authors because it's sort of a case study. And as you can see, it's just kind of heavily weighting the left-hand side there where the full models are kind of being run with the least amount of weight on the flow doesn't do anything hypothesis. All right, so I'm just gonna go straight into the results. So first of all, I'm gonna go over the water cost results from calcium. In the x-axis is the different alternatives, and the cost is in 1,000 acre feet on the y-axis there. You can see that if we don't do any action, obviously it's not, that's kind of the baseline. We said that there's no cost for that one. You see that there are a couple of alternatives with moderate costs. So that's the black and the greenish color. So that's the fall status quo action, and then the summer only. And then there are a couple of actions that are kind of really high costs. So that's kind of the high flow in the fall at 74 kilometers and the combined summer and fall. The results for the delta smelt is a bit different because we have, again, 8 variants of the model. So I'm going to walk you through this. So this is kind of showing us the influence of uncertainty on the ranking of alternatives. So the y-axis is the lambda. Again, the x-axis shows the different alternatives. So I'm just going to highlight one at a time. So this is about where no food or movement change based on flow. As you can expect, there's no meaningful difference across alternatives. You're just getting noise, right? Everything performs equally good or bad. Well, if we just allow movement of delta smelt based on flow, but we don't allow food to change, then it seems that summer fall action performed best for delta smelt, quickly followed by fall X2 at 80 kilometers. If we keep the food component only and we keep delta smelt distribution static, then the alternatives that include summer action perform best. So that's the one in green and the one in blue. So that's the summer, fall, and summer only. And if we keep the full set of models, we allow food and movement to change, regardless of which IBMR version we use, the summer, fall combined action perform best. But it also is a bit different depending on which version we use. The one with the daily time step shows that summer action is really close to the combined action compared to the version 1. Okay, so given all that, what is the best decision to make? So I'm gonna spare you all the math, so I'm just gonna kind of explain that we calculate utility score, which is kind of a way for us to combine these two metrics, right? We wanna kind of maximize delta smelt while minimizing supply costs, water supply costs. So we have this score to kind of get a sense of like what would give you the best happiness, sense of happiness for the decision maker. And for those of you who are familiar with decision analysis, you can ignore this if you're not familiar. I use the local scale to kind of evaluate just the range of outcomes in this decision context. And we use the hypothesis weights that I kind of previously showed based on our set of co-authors. But we allow objective weights to vary. It's simple because we only have 2 objectives here. Okay, so to just kind of show this, this is the output that we have. So on the x-axis is delta smelt objective weight. So this is sort of how much you care about one objective over the other, given the range of values that you see. So on the right-hand side is if you're at 1, means that you only care about delta smelt. If you're all the way to the left, it means that you don't care about delta smelt at all. You only care about maximizing water supply. The y-axis is kind of this sense of happiness, this utility score, and the lines are the different alternatives. So, wherever you are on the x-axis, you want to pick the ones with the highest utility score, the line with the highest utility score. So if you are a decision maker and you care more about water costs, then the do nothing or no active action would probably be chosen. You would want to choose that. If you're somewhere in the middle here between between 0.5 to 0.75 delta smelt weight objective, delta smelt objective weight, then you would probably choose the summer action, and you would go all in with the summer fall combined action if you really, really care about delta smelt and less so about water. Okay, well, what about value of information? So again, this is the benefit of waiting before making a decision by gathering information so that you can know for sure which one of these hypotheses is correct. And this gives you a sense, right, like whether or not like there's value and how much value there is in kind of undertaking a study to determine which one of these models is correct. So if you do the calculation, again, I'm skipping all the math, but I can show you later during the break or something if you would like. You can see that there's 0 value if you care more about the water cost objectives. Generally, there's only value in studying this subject further for the decision maker if you value smelt a bit more than water supply. The other thing that we can do with this is we can break it down to value of partial information. Remember, we have 3 areas of uncertainty. we can kind of look at it on an individual by individual basis about which uncertainty you want to resolve the most. So in this example, you have bioenergetics, food, and movement. But as you can see, the blue line is always on the top for the most parts, meaning that in this particular case study, if you want to prioritize research, it should generally be more on zooplankton or food. Specifically on the seasonality and spatial variation of how zooplankton changes to flow, with flow, that would really be informing the IBMR. So something that works with the delta smelt model. But note that this also changes depending on the objective weight, right? So if you really, really care about delta smelt, then you actually would prioritize understanding delta smelt distribution changes based on flow more than food. And when you get towards this right-hand side of the graph. All right, just to summarize, I think we've created a tool here that would allow us to make a more informed decision on X2 action. It was way more simplified because there are a lot more things that we care about when we make this decision in reality. But I think it's different from other studies so that we kind of think about more than one objective, right? In a lot of studies, we think about like, oh, well, how can we maximize salmon? How can we, maximize the smelt? But this is kind of like thinking about it in multi-objective framework. And O'Connor also shows us that, the values of decision makers really have a strong role to play on all this. It determines how much value doing more science, is, whether or not there's value and how much there is in doing more science. And it also changes, how you would make which decision you would make. And it also shows us how our alternate beliefs and how the system works affect the decision. And I just want to note there's a lot of details that I'm leaving out with model assumptions and caveats and whatnot, but I just don't have the time. So keep that in mind. With that, I'd like to thank all the folks who have provided input, especially Sam Bashevkin, who originally constructed the salinity zooplankton model. And I'll leave you with this. I hope the Shiny app still works. If you scan that QR code, it'll take you to the Shiny app that I built for the study, where you can be the decision maker in this case study. If you go to the swing weighting, it'll kind of tell you how you can get your personal weights on the different objectives. And then you would go to the line plot to just kind of see what decision you would make if you were the decision maker. And with that, you can take questions.
We've got time for a few questions.
Hey, Brian, thanks for sharing this. So you have kind of a scenario where a decision maker has particular weights on smelt versus water cost, but frequently in this system, we have a group of decision makers who have different weights on those costs. And how would you suggest this translate into a group decision-making context?
Yeah, that's a good question. I don't think I wouldn't consider myself as an expert decision analyst. I think there are different approaches that you can take with that, but that is certainly not typical. From talking to Mike Runch and Brian Healy, who are kind of like the folks who have been doing decision analysis for a much longer time, they noted that like this multi-decision maker setup is not really common, when they've worked in the East Coast on whooping cranes and in the Grand Canyon and whatnot. So that makes things a bit more complicated. There are approaches that you can take to do that. But yeah, it is not like something that's going to be easy to do, and it's not as straightforward, that's for sure.
Let's take a question from online.
Laura asks, can you explain again what the Sassoon Marsh salinated control gates action is under the 8 scenarios?
I believe it's the 60 days constant action whenever the salinity is triggered at Belden's Landing. So the calcium, one of the calcium output is Belden's Landing salinity. It's in it's in conductivity, you see, but we translate it to practical salinity unit, which then gets plugged into the salinity zooplankton model. And then it'll tell you like how much zooplankton you would expect.
I'm just curious if the water costs that go into your model incorporate the potential for costs of using any water to increase as wet years or below normal years become more rare due to climate change? Is there any flexibility for that in your model?
Yeah, I'll try to understand. Yeah, I think I understand the question. So to clarify, like the action only occurs in wet and above normal years in this scenario, because during drier years, there's just not enough water to kind of go around. Typically, we don't do that in reality. Does that answer your question? Yeah, so it's the way water cost is calculated is based on how much south of Delta export would be reduced, and also how much additional water would have to be released out of reservoirs.
Okay, yeah, that's yours. Thanks.
All right, thank you, Brian.
Thank you.
All right, next up, we have Kim Luke from Delta Stewardship Council with her talk entitled Science Action Agenda Progress Summary.
All right, hi, I am Kim Luke. I'm an environmental scientist with the Delta Stewardship Council, and today I'm giving a lightning talk on the Science Action Agenda Progress Summary. The Science Action Agenda is a document developed by the Delta Stewardship Council that outlines priority science needed to address management needs and to address knowledge gaps. It's developed publicly, collaboratively, and it's updated about every 5 years. And we're currently on our second full iteration of the Science Action Agenda, which is the 2022 to 2026 Science Action Agenda. And there is a QR code up there if you'd like to scan it, and you can read the document there. The Delta Stewardship Council uses the Science Action Agenda several different ways, one of which is along with the Delta Science Plan and the State of Bay Delta Science. The SAA is a part of the Delta Science Strategy. And this is intended to guide and benefit the Delta community. And what else? Yeah, and establish the foundation for One Delta, One Science. We also use the SAA to guide funding within the Delta to ensure that research addresses our priority management needs. And with our first full iteration of the Science Action Agenda, the 2017 to 2021 version, we guided over $35 million in science investment funding through competitive research award processes and targeted actions. And this was done with the Delta Science Program, along with our partners at US Bureau of Reclamation, the California Department of Fish and Wildlife, and state water contractors. To give you a look into the current Science Action Agenda and kind of the structure of it, I have an example of one of our management needs. So this is management need number 1, which is to improve coordination and integration of large-scale experiments, data collection, and evaluation across regions and institutions. So this is our overarching management need. And then under that, we have our specific science actions, which are those public and collaboratively developed ways to address that management need. So here we have 3 different science actions. And I just want to note that the little green bricks on there for A and B indicate that these are science actions that build upon the previous iteration. And C has a little sun next to it, and that indicates that this is a science action that's unique to this new iteration. And again, these are intended to guide research in the Delta. So with something like the Delta Research Awards, proposals will have to identify which science actions they're addressing. Right now, we are conducting a progress summary of the current Science Action Agenda. And the purpose of the summary is to document and evaluate progress. And we do that by evaluating all the individual science actions. This progress summary also informs the next iteration of the Science Action Agenda. And it gives us, a sort of gauge of return on investment, because we can see how the money we're investing into research is helping us achieve our goals in the Delta. And for this progress summary, we use both a qualitative and a quantitative approach. This is an example from the progress summary of our first full iteration of the Science Action Agenda, the 2017-2021 version. And this is showing a quantitative approach. So for a specific science action, which I have outlined in a red box, we're looking at all of the science activities that are addressing that, which is the other red box in that plot there. And then we break it down by type of project. So whether it's funded research, modeling, a review, and then we assign a status to that science action. So for this one, it has significant progress. And in our qualitative approach, we kind of go more into depth of how we got to that assignment. So as you can imagine, it's really important that we really have the breadth and depth of research going on in the Delta. So we have a really robust data set to evaluate our progress. And that's where you all come in. Of course, we have our own funded projects that contribute to the progress summary that we can track, but that's really just a small piece. As we've seen these past few days, there's so much research going on in the Delta, and we want to know all about it. So for our current progress summary, we're planning to use the Delta Science Tracker as sort of our one-stop shop to see all the research going on in the Delta. And there are benefits to you as well for adding your project to the Delta Science Tracker. I have an example of a landing page for a project here. So you can see the description of the project. You can see collaborators, potentially the region that the research is going on, as well as any products that the project develops. And one really important feature on here is that you also get to see the number of views. So you can see how many people your research is reaching. Another really awesome tool in the Delta Science Tracker is that you can use it as a search engine. You can look for new collaborators, you can look for other projects, and you can summarize and create visualizations like the one I have up here showing different collaborators in the Delta. So, you know, again, there are a lot of benefits to you for using the Delta Science Tracker, but it also is an opportunity for you to contribute to Delta Science at a larger scale and help make the Science Action Agenda Progress Summary the best it can be, which will in turn make the next iteration of the Science Action Agenda the best it can be and help us reach our management goals in the Delta. So thank you. That is my lightning talk. And I will take questions if I have time. Also, there's a QR code for the Delta Science Tracker if you want to scan that. Great. You can find it.
Thank you, Kim. All right. Next up, we have Andrew Veery, subbing in for Kai Ross from the Kramer Fish Sciences. His talk's entitled Machine Learning for Natural Resource Management.
All right, good morning, folks. Like you said, my name is Andrew Veery. I'm the image analyst with Kramer Fish Science. And let's go over machine learning for natural resource management. So what is machine learning? It's a method where computers learn patterns from data to make predictions or decisions without being explicitly programmed for every step. And they're very often built as supervised classification, where already classified training data is used to help predict or classify new data. I'll take this slide as a point where I could help differentiate machine learning from AI. A lot of people get interchange the two frequently. Machine learning is, as it says here, the step in which the computers make the prediction from patterns it's learned, where AI is more of the step back, the general tool. Use the example of programming a robot to do a human's job. If the robot wanted to pick up red blocks, the camera would feed into the machine learning model which block is red, and that would tell a robot to pick that block up, where the AI is use a lot of the electrical engineering to control the robot as well as the model used to predict, right? Just an outline of my talk here. I'm going to go over the brief pros and cons about machine learning. Machine learning examples from the least complex to the most complex. We have at the start an old tool, regression models, which a lot of people are familiar with. And then we get into slightly more complex pattern recognition models. And then eventually we'll get on to talking about deep learning for computer vision. It's also worth mentioning the size of these blocks. Deep learning is maybe 1 million, billion, trillion times more complex than a regression model. But the artwork here is just to help illustrate this a little more simply. And we'll go over some best practice tips as well for implementing machine learning yourself. And then a quick summary at the end and time for Q&A. So the pros and the cons. The benefits, one of the major ones is data driven. So the machine learning model, the model is derived right from your data, right? It's not using any intuition or, external thoughts. It's not making assumptions. It's based off of entirely what you feed into it, right? So the preparation in your data sets, the cleaning, the reliable sources, that's all extremely important to a successful machine learning model. They can also model many types of relationships, whether that be linear or nonlinear, few features, thousands of features. And once they're built, they're easy to implement into analysis pipelines, and they provide extremely rapid and consistent outputs. That's an also extreme benefit of machine learning models, right? Where we are in the world, the amount of data that's ingested, across all fields on a daily basis is extreme, and it's growing every year. I think the number is, they call it a zettabyte. It's a trillion gigabytes is what expected as the amount of data that's recorded every year across fields. And that's just growing every year too, as technology advances. We implement these new sensors, they get more advanced, take more pictures, more high resolution, and it just adds up every year after year. So we need these pipelines to, efficiently process this data. Cons, however, being so data driven, it requires, like I said, a lot of training data. It needs to be well labeled by professionals. It's time consuming, but in the end, it's extremely worth it. And they're often considered black box approaches, where you can't exactly explain what's happening during every step when the model is making a prediction, depending on how complex it is. You lose a little bit of transparency there. So you need to, do a lot of validation and analysis and statistics to help prove something's working responsibly. And there's also issues with edge cases. So something that you weren't expecting popping up when during your training data, right? So that 1%, that 2%, those outliers that you don't expect to happen so often, you might have missed before you're preparing your training data set. You need to be wary of those, and you need the validation. Like I said, so those two kind of coincide. You don't want to go predicting on months and months of data and then not go back and cross-check stuff, right? Because it can make these slight mistakes, and if you don't pick it up and notice it, they might slip through analysis and eventually a decision, which is what we want to avoid. So the regression models, if you're not familiar with these are continuous predictors that result in a continuous variable. They assume linearity in your data set. And these can be used in management for feature selection here. We have one of the tools is the least absolute shrinkage and selection operator, or lasso regression. What that does, if you've fit regression models in the past, you've had issues with overfitting. So your model performs really well in the training data set, but then when it goes and predicts on data not seen during training, it underperforms, also known as overfitting. What you can do is it's called a method called regularization. It adds these penalty terms to your residuals. So I think that the regular regression just uses the sum, whereas the least absolute uses the sum of the absolute values. So what that does is when it's training, it shrinks your features, the importance and the coefficients to 0, which in the end lead you to know that those features weren't important in predicting the variable that you're interested in. It'll result in also coefficients for the ones that you might be interested in. So you can see where to allocate resources, right? If you have $1 million to spread over 10 areas, you may not want to spread it thin across each, because some areas might be more important than others, and others least. So you can use something like this in order to help, allocate resources. The random forest model is an example of an ensemble method. It's unlike regression, it's robust to nonlinear relationships, but we start to get, we start getting a little black boxy, like I said. So what a random forest is, if you're not familiar with it, it's just an ensemble of decision trees. A lot of people know what decision trees are. You start kind of with a simple where your data is most separable. You start there in your tree and you kind of dichotomously go through the tree at your different nodes and you pick yes or no, yes or no, yes or no. And eventually you lead to a classification. You can also use these for regression, as you see here. So you can predict continuous variables or you can classify with it. But this also supports variable importance too. So you can get that same idea out of it of what you should allocate resources to or not. But like I said, it's robust and nonlinear relationships. And then we get a little more complex here with gradient boosting. So unlike random forests that, you know, with each decision tree in the ensemble, it uses a lot more random. Gradient boosting is a little more Bayesian in its methods, where it takes the error from your first tree and bases the parameters of your next tree off of that. So your decision trees sort of learn from the last model in the training until you eventually reach your result. These are the same thing, very robust and nonlinear relationships, but they're computationally more expensive. You need a better computer or you need more time, and it requires more tuning. And as I said, high accuracy, and they help you determine variable importance. Next, we have pattern recognition. So we're not just predicting individual numbers. We use sequences in time or arrangements of values, images. We see here it's a spectrogram. So we have a value on your Y, and then over time, how it fluctuates. We can learn something from that pattern. We'll go over soon. And then we have an image in an XY. So we have just two directions for our data there. Nothing, time's not involved. And then we start getting into spatial data, right? So we have colors, more shapes. Might have more layers, more bands. Maybe some of you who are into birds are familiar with the Merlin Cornell app. This was built, sorry, I forgot the organization, but these are spectrograms to help identify the bird species from their songs, right? So this is a very impressive app that just works with your phone's microphone, right? And it's going to access the model on your phone and use your phone's processor to compute this, right? So this works extremely lightweight. And from who I've spoken to about it, it works very well. I haven't tried it myself, but maybe some of you in the crowd have. What that does is takes time series data, so the frequency of the noise it's reading over time, and in a certain frame, it's able to tell you at a good accuracy what bird that song belongs to. Binary arrangements, so OCR, optical character recognition. This is used to help sort of convert text data into computer readable strings. So PDF converters, you see here, this is an example. This page is from 1971. Back then they didn't have PDF converters. Somebody probably had to type this up on a typewriter, into a database. Now you can just scan hundreds of millions of documents, thousands of documents, and then you can sort of use programming techniques to tell where the text is. And then your character recognition is going to tell you what each letter is, where there's a space, and it'll build you sentences there. Next, we have complex arrangements. So people who are GIS experts, you might be aware of this. Geospatial object-based image analysis. So we have an image, all those images are the same area, but we have multiple bands, right? So each of those bands could, correspond to different wavelengths emitted. They could correspond to different, types of land use, types of land cover, or that's what we're trying to predict, right? So we have 6 bands there, and your complex, we use here convolutional neural networks. They'll scan the image and compare it to the training data used from the model, and we could probably tell where the trees are, where the roads are, where the land is. And this is extremely helpful when it comes to land use cases, land mapping, monitoring deforestation, fire risk, and you can combine this into time series too. So you can take all these bands and you can look at them over weeks and see how they change, and you can model that too, just to help detect risks. Now we're getting to the most complex. It's going to be deep learning for computer vision. The image composition includes shapes, colors, textures, symbols, and these complex models consider these aspects to see the images, allowing you to detect where the object is. So we have in this example, it's a little fish. It's going to detect where it is, and it's going to give you a classification. You can do a segmentation problem. So it'll tell you not only a box around where it thinks the fish is, it's going to tell you exactly the pixels that belong to that fish. So you can get areas, you can get boundaries, perimeters, shapes. From that, we can make automated measurements. So that's getting the fork length. It's just the longest distance between 2 points in the mask. And then that height is, a line perpendicular. So, and then we can track. So we can use an algorithm to reassociate over time, a frame with its previous frame. It's able to see, with different distance metrics, that fish is actually the same one that I made a detection on from the last frame. And over time, where it would have counted 10 fish here, we only know there's only 1 fish. Let's go over a few applications. Like I said, from the last slide, detection. What we have here is a shadowgram of water flowing past the camera. It's a very small camera and it emits light. When the light comes back, it graphs these shadows essentially. This is extremely noisy data, but we're allowed to see the bugs amongst the bubbles and debris. Right now, this model pretty accurately classifies copepods, sclerocerans, and amphipods. You see there are all those black boxes, a lot of those highlight noise and false predictions, but the model will then iterate over boxes and tell you bug or not bug and which bug it belongs to. And we have there a pretty little copepod. But you can see all the noise behind it, right? So we got to smooth that out, blur that out, filter it out, and get it ready for prediction. In our sample days, we can collect somewhere between 50 and 250,000 of these images. So you can probably assume how long it would take to manually view that. It's very difficult. You need to zoom in, find bugs, maybe take a week. Whereas this model, I can run this in a few hours. It's extremely efficient. Up next, segmentation and measurements. So we have here some substrate with a scale bar. What the model is going to do, same thing, it'll iterate convolution over the image and return to us which pixels it thinks belongs to each rock. Like so, it gives each rock a unique mask, designated by a different color. And then what it'll do is find the maximum distance between 2 points, and that's your A-axis, your major axis, and the line perpendicular to that is your minor or your B-axis. And then the scale gives you a ratio of pixels to centimeters or millimeters. And then you export your data set and you have a substrate analysis. And then on to this last example, combines most of these techniques into one. So that fish lacks the box, but the box was detected. And then key points are drawn. So this model was trained to tell where on the fish each key point was. Key point here is our nose, eyes, gills, adipose, a few on the anal fin, the caudal fin, and the pectoral fin. So we can use that to sort of fit a fork length better than just drawing it across the whole detection box. This fits it a little better, and we can sort of build trusses to help maybe in another classification instance. And those are also tracked. If we had the time series data there or a video, you'd see that fish being successfully tracked. Considerations, you need pre-trained versus custom weighting, so that's hardware needs. So pre-trained is just like a public model online that you get that you might not be custom fit to your data. So you need to take that into consideration because training these models takes a ton of computational power. Be wary of the edge cases, like I said at first, those 2%, 1% cases that you don't see during training, you can be wary of those. And then save your data for both training and validation. So data that your model is predicting on, save those results so that in the future during retrainings, you can use that or you can set them aside for testing and validation. So just a summary, machine learning, AI, it's buzzy, it's broad, it's really useful. People here, they want to know how am I going to implement this projects? It's great at scale. Like I said, speed and the consistency. Consistency is a good thing to mention. It's going to predict in the same way every time. It won't get tired. It has no emotions. It doesn't have a good day. It doesn't have a bad day. Just it's running right now on my office while I'm here. But you need data. So a ton of it. It's some of my examples I give for computer vision models. Some of my models have 30,000 images, 40,000 boxes drawn. You need a lot of data. All the different cases of your objects. It's really important that you have, your trained data itself is robust. so that the predictions can handle all these fluctuations of different instances that's thrown at. And then it needs validation. So just like I said, predicting on a large amount of data without going back, cross-checking, spot checks, fluctuation in results that, are outliers. You say, why did it do that? Go back and check it. Pretty simple. And it's the coolest part, it's just going to get stronger. So since I've gone into this machine learning, these models have improved quite a ton, and especially with how fast they are too. Some of these models increase in speed tens of thousands of times, and the computers that you use to predict with, they're only getting better as well. So that's good. And then I will wrap up and ask if anybody has any questions. I don't get my glasses.
Time for a few questions.
He was first.
Right there. Excellent presentation.
Thanks.
This is really exciting to kind of see the technology evolve. So it's clear to me, it seems that the potential is there, but I know, working with things like beta remote underwater video and doing individual identification in the past, when you're relying on humans, things like max N are a problem where you're only able to confidently speak to abundance in a single frame to avoid overcounting, duplicate individuals. Is that something that these algorithms are currently incorporating? So if you're looking at a fluid stream of video, that those measurements can actually avoid duplication. Those measurements seem like they'd be potentially able to do that. But if you're really concerned about abundance and you have an hour of video, is that something that's currently being taken into account? Is using individual morphometrics to eliminate double counting?
So what you're asking is how successful are the tracking algorithms and not counting an object twice in the video in consecutive frames?
Correct, yeah.
Yeah, so that's, to get quickly into the math, you use stuff like the velocity. You can track which way it's going in, and you see those boxes drawn over the fish. What the algorithm will do is project into the next frame a box, and then based off of the detection of the next frame, how much overlap it has, and then it uses distance metrics related to color or patterns from the deep learning model to extract two. So you can make a distance. You can make distances from like spatially. With your Euclidean distances, how far it's traveled, and you can also compare the colors and the shape.
But I guess the question would be, say, you have 30 minutes of video, 15 minutes later, an individual that had already been captured re-enters the frame. Right. Can those algorithms differentiate between that?
So that's an issue. Yes, that's an issue. So what I'll do, for example, in the fish ladder case, if I'm confident in their system that fish love to travel past the camera and not come back, you don't got to worry about that. But what I'll do with where the tracking algorithm starts, if it's X coordinate here, when I lose the track, if it's positive, it's traveled this way, right? If it's negative, it came a little bit and head back, so I won't count it. That's something I'll do to combat that. Unfortunately, if a fish heads up the fish ladder for 10 minutes and comes back, that's a difficult case. We could save it. So I could detect it here and then notice, it's coming in from the left side of the stream, just using an X coordinate, right? And then you print that out and have someone validate it. Say, hey, go look at where it came in. This fish came in from the left side of the box. Either don't count it or go validate it, see maybe it's a fix that we've already seen. There are techniques, but that's something that is, a tough to handle issue. I agree.
Great, thank you.
Yeah. Go ahead.
Great, thanks for the overview. Yeah, by the way, the app is Merlin from the Lab of Ornithology. You were wondering about that. The question I have is about data. So a lot of data doesn't necessarily mean representative data or high quality data. One of the examples is a lot of the heart disease analysis is done on old white men, right? So if we were trying to use that to do some other kind of prediction outside of that group, it would be very difficult. So can you comment on that, making sure the data is representative of what it is you're wanting to do? Thanks.
Yeah, sure. So it sounds like this is, this is kind of what you take into consideration when you're like a data scientist for a specific organization. So what a data science does is combines like three things. You have the domain, so health science domain, and then it involves programming and statistics. So in the domain, if you're not sure, where this health data is coming from or who it may apply to, you need to collaborate closely with a professional who can help inform you. And it's a lot of collaborative effort to handle stuff like that. If you hired me and I right onto that project and I didn't know how a lot of the data, was gathered or who it was, important to, I would need to closely collaborate with an expert who did. And with my programming and statistical skills and their knowledge of the field, we could help to work together to model what it is you're looking for. Yep.
Yeah, data, the validation of species, for example, is part of data quality, as is image focus, degree of focus, and the performance of the models really decreases quickly with loss of focus.
Right.
But I've just been learning about something called multi-factor learning. And it makes me realize that you can take a library of highly focused
right.
and then use a lesser quality camera and transfer that learning to, and it makes me wonder, especially talking to Kramer, with, just the possibility of like a Bay study, a Bay Delta net kind of thing where multiple libraries are available for, the public to use. I don't know if Kramer would be into that, but I think it would, there's a lot of value of thinking about how to create something like that.
So like a public repository of
Exactly, of images that.
of images. Okay.
Yeah.
That's a good idea. If you want to reach out an email for, more on that topic of creating new projects or more details, but what you're saying about the image enhancement, it's correct. Yeah, the quality of the image directly affects how easy it is to build a model to predict on a model with, similar data. Image enhancement from deep learning is a thing too. So you can take a lot of blurry images and you can train it on what the image is supposed to look like. So in the future, you actually pre-process an image with a neural network to make it look prettier. And then that'll prepare it for another neural network to then help what I went over here to help classify and everything else. Yeah.
All right. Thank you, Andrew.
Thank you.
The last talk of the session today will be from Shruti Khanna from the IEP with her talk entitled Landscape Scale Multi-Year Evaluation of Floating Aquatic Vegetation Control, 2004 to 2018.
Thanks, Nick. Yeah, arrow keys. So hello, everyone. So today I'm going to be presenting the results of our investigation of the effectiveness of chemical control of floating aquatic vegetation, or FAV, in the Delta. I'll call it FAV so you guys don't get confused. This study is unique in its temporal and spatial scope, incorporating treatment information going back 2 decades for hundreds of sites spread across the Delta. The study has benefited from many investigators over the years, and the 2 I have listed here are just the ones without whom the study would have been impossible to complete. One major data set that made this work possible is likely the longest existing spectroscopy data set in the world. Imagery of the Delta has been captured by airborne spectroscopy sensors every year since 2004, except for a data hole from 2009 to 2013. The Seastars lab at UC Davis, run by Dr. Houston, has been responsible for classifying aquatic vegetation using this imagery and publishing it for public use. For the purposes of this study, we used class maps from 2 time periods, 2004 to 2008, and again from 2014 to 2018. Treatment data from 2001 to 2018 was made available by the Division of Boating and Waterways at state parks. We selected 461 sites as treatment and reference sites. These sites were chosen in 3 different kinds of habitats: main channels, shown in blue, flooded islands, shown in red, and slow shallows, or the shallow regions between small remnant marsh islands within a channel, shown as green here. The reason we examined the efficacy separately in these 3 habitats is because they are hydrologically distinct. The flooded islands are shallow and have higher water residence times and low flows. The channels are deeper, have higher flows, while the slow shallows are shallow, but likely still have low water residence times because they are completely open to tidal fluxes. For the purpose of this study, sites in dead-end sloughs were also lumped together with slow shallows. Over the period analyzed in this study, FAV was treated using 2 herbicides, 2,4-D and glyphosate. We had a natural experiment on our hands because in 2004 to 2008, as you can see here, the dominant herbicide in use was 2,4-D. While in the latter time period, 2014 to 2018, the herbicide glyphosate was the dominant one. To test the efficacy of one independent of the other, we filtered our data set to those sites exclusively treated for 2,4-D in the first period and exclusively treated by glyphosate in the second period. We used a generalized linear mixed model approach because we needed to account for both fixed and random effects due to repeated measurement through the years per site. We developed 2 sets of models, one to test the effect of an herbicide within the year of application, and one to examine the legacy effects of the herbicide 1, 2, or 3 years post-application. We had 2 sources of information. From DBW, we had the vector paths of the boats spraying the herbicides and the daily logs of which herbicide was sprayed in which reach scale region of the delta. Also, when and how much. I can see the mouse not there. Yeah, sorry about that. From the class maps that we generated from our imagery every year, we had the location and extent of every FAV polygon or site for every year of the study. We use this information along with the boat paths to create the 461 sites that I was talking about that were treated and referenced. Finally, we computed the following covariates for each polygon or site in our study: the herbicide sprayed per unit area, the spray frequency or the number of visits within a single spray season, and continuous years that that particular site was sprayed. A response variable, because this was a logistic regression, was the probability of FAV occurrence. The next 2 slides will show the results for 2,4-D used in the 2004 to 2008 time period and glyphosate used for the 2014 to 2018 time period. Results in this case refer to the simulated values of FAV probability across all values of all covariates as they appear in the original data set, generated using the effects package. But let me explain first what these panels will be showing. The 3 different columns will show the results for the 3 different habitats: channels, slow shallows, and flooded islands. Each plot will show the probability of FAV occurrence on the y-axis and log to the base 10 of the amount of herbicide sprayed per unit area on the x-axis. Values predicted for the sites with higher or lower spray frequencies are plotted separately. The 70 to 100 percentile spray frequencies, and by spray frequency, I mean the number of times that a site was visited within a single spray season. So not across years, but within the same year. So the higher percentile spray frequencies are in orange and the lower percentile spray frequencies are in blue. Finally, the top row shows the results for sites sprayed only 1 to 2 years, while the bottom row shows results for sites sprayed more than 6 years consecutively. Site and year are considered random effects because we had data from each site for multiple years that are treated as repeated measures. Okay, so let us look at the results from the 2004 to 2008 data only for sites treated with 2,4-D. The gray dashed lines show the probability of distribution of the reference sites. As herbicide application per unit area increases, we can see that the probability of occurrence of FAV decreases. In channels and slow shallows, the herbicide is only significantly effective at higher doses. But in flooded islands, the herbicide is effective at a lower application rate. The effect of higher spray frequency is harder to see in slow shallows or flooded islands, but clear in channel habitats. It appears that it is better to spray a higher dose of herbicide less often than to spray a lower dose more often within the same spray season. Spraying a site consecutively for many years does not seem to confer any advantage to the site. Most of the time, it seems to be less effective. This might simply be due to the fact that sites that are sprayed year after year might be persistent sites that already have a higher concentration of overwintering organs and maybe nursery sites. So in effect, 2,4-D seems to be the most effective in flooded islands, somewhat effective in slow shallows, and least effective in channel habitats. Now let's look at the results from the 2014 to 2018 data only for sites treated with glyphosate. Similar to 2,4-D, as glyphosate application per unit area increases, we see that the probability of occurrence of FAV decreases. However, with glyphosate, we see very clearly in both slow shallows and flooded islands that lower spray frequencies with higher concentration of herbicide per visit is far more effective than the opposite. In fact, a significant reduction in FAV probability is only observed when the spray frequencies are lower. Similar to 2,4-D, glyphosate seems to be least effective in channels and most effective in flooded islands. It does not seem to do better than 2,4-D in slow shallows, but keep in mind that all of that is true only at lower frequencies of spraying. So what you see here is that there is significant reduction in FAV occurrence only at when you spray less often, but spray more herbicide per visit. The second set of tests that we did were to determine if the impact of the herbicide could be detected 1 year, 2 years, or 3 years after treatment. In other words, were there legacy effects of the herbicide treatment? For all sites treated in the year Y-1, but not treated in the current year Y, we propagated the continuous years of treatment variable from the previous year to see if the treatment history of the site had an impact on legacy effects. Now, we did not do this for the amount of herbicide sprayed per unit area or the spray frequency, because that only applies, like, what year are you going to bring that information from? Let us say that the site was treated 3 years consecutively. Every year, it might have been a different amount of herbicide and a different spray frequency. So the only variable that we investigated here was the continuous years of treatment. And we only did it for 1 year later because that was a reasonable sample size. If we look at 2 years before or 3 years before, we just didn't have enough sample size to do that. Okay, so what you see here, and I'm going to show you the results for glyphosate in this case first, and you'll see why. In channel habitats, we see that continuous years of treatment has no impact on legacy effects. In fact, the sites that are treated 1 year ago were no different from reference sites, no matter how many times the site may have been treated before that. However, in slow shallows, A site treated for more than 6 consecutive years showed a significant legacy effect 1 year later. In flooded islands, sites that were treated for more than 2 consecutive years showed a significant legacy effect. Thus, legacy effects were present for both slow shallows and flooded islands, but they were stronger for flooded islands. Now, let's look at 2,4-D. We see that in channels and slow shallows, the continuous years of treatment have a negative impact on legacy. The longer a site has been treated, the more likely it has a higher probability of FAV occurrence a year later. And in flooded islands, the treatment history is irrelevant. Thus, it appears that the legacy effects of 2,4-D are either neutral or negative. Replacing the continuous years of treatment with a simple flag for not treated or treated 1 year ago, 2 years ago, or 3 years ago, let's see how long these legacy effects actually do last. So again, let's look at glyphosate first. We see that in both channels and slow shallows, there is no significant legacy effect. Now, remember that the results for this are slightly different than that when we included CY, where we did see an impact in the slow shallows, and that's because it's a different variable. It's a flag, right? So it's going to have slightly different results than using the different covariate. Okay, so both in channels and in slow shallows, there is no significant legacy effect. Treated sites are not significantly lower in FAV probability compared to reference sites. Only in flooded islands, we see legacy effects for 1 year later. Not only just 1 year later, but 2 and 3 years later too. This I thought was pretty amazing, not something I was expecting. The N that is quoted here above the distribution is the number of sites that we had that satisfied this condition. Keep in mind that when we are looking at legacy effects, we are already down to all of the sites that were not treated in this year. We are only looking at sites that were treated 1 year ago, 2 years ago, 3 years ago. So naturally, we have a smaller sample size. And it gets smaller and smaller when you're looking at 2 years ago, 3 years ago. Okay. So taking all of the previous results into account, one can conclude that flooded island habitats are probably the easiest to treat and sustain the effect of the treatment. Now let's look at 2,4-D. We see that in all 3 habitats, there is no significant legacy effect. Treated sites are not significantly lower in FAV probability compared to reference sites. This also was kind of surprising for us. Taken all together, it appears that the results are showing that while 2,4-D appears to be mostly as effective as glyphosate in the year of application, but it is not great when you look at legacy effects. It doesn't have any. So all of this can be distilled down to what is important for adaptive management, what is important for actually treating these FAV species. First, while both herbicides seem to perform comparably in the year of application, glyphosate has staying power, but only in flooded islands, right? Treatment is most effective in flooded islands, somewhat effective in slow shallows, and mostly ineffective in channel habitats. And I don't know if you noticed the raw values on the y-axis for the probability of FAV occurrence, but there are 2 facts to be gleaned from that. One is that the general probability of FAV occurrence in any of the 3 habitats was much lower in 2004 to 2008 compared to 2014 to 2018. The axis actually on one of them goes to just like 0.3 and the other one goes to 0.8. So there's quite a bit of difference. The other thing is that within those 3 sites, those 3 kinds of sites, channels anyway have the lowest probability of FAV occurrence because of high flows and the kind of environment that FAV likes to grow in, it's least likely to be in channels anyway. Right? So considering all of those things, maybe it is not worth to treat channels. This is what at least the results seem to be showing. Okay, so in terms of frequency of application, this was also interesting to see that it is probably more effective to treat a higher dose of herbicide in a single visit than to visit that site multiple times, but treat it with a lower dose of herbicide. And this was clear to see, especially with glyphosate. Finally, continuous years of treatment do not seem to lead to better control. However, in the case of glyphosate, we saw that just in flooded islands, they did seem to confer some advantage to lasting legacy effects of the herbicide. So we also strongly advocate for consistent monitoring and setting aside of reference sites in addition to treatment sites, which is a must for evaluating this kind of efficacy. Without the availability of these long monitoring data sets, such an intense spatiotemporal study of herbicide effectiveness would be impossible. Success must be defined a priori with regards to both primary objectives such as reducing cover to secondary objectives such as improved water quality, and then examine whether they are fulfilled. This study, for the study, we would like to acknowledge the help of the staff and students of the Sea Stars Lab over the period of the study and over the period of all of the years of imagery that were taken. We would also like to thank the State Parks Division of Boating and Waterways for sharing their detailed treatment data with us from 2001 to 2018. Lastly, we'd like to thank Louise Conrad for her mentorship, guidance, encouragement, and stewardship of this project from its very genesis. Thank you.
We got time for a question or two.
Yeah, Michelle?
Was there much of a difference depending on the species of FAV that was treated?
So there are two main species of FAV in our period of study that were treated. That was water hyacinth and water primrose. Now, water primrose, they only got permission to start treating around 2016 and probably started treating around 2018. So it would only apply to the later time period. And it becomes very complicated with the fact that, let us say you had water hyacinth 1 year, you treated it, and the next year it's water primrose, and now you're treating water primrose. We decided, at least with this initial study, to not get into it. Like, okay, let's look at general efficacy. I think as the data set fattens, because we do have imagery since 2018. We have had imagery taken 2019, 2020, 21, every single year until now. If we got the treatment data for that, then we'd probably have enough to investigate the efficacy for each species separately. Now there's a third one, there's alligator weed, which they also treat. So that's 3 species.
Yeah. Let's do one online question as the end.
Yes, we have an anonymous question. With higher concentration applications of herbicides, is there any concern with impacts on other species in those environments?
So that is a question, I'm sorry, I cannot answer. I'm not a toxicologist, but that is a very relevant question. It's very easy for me to say that is more effective, but whether that is permitted, allowed, good to do, I am not the right person to answer that question.
I would say yes.
All right. Thank you, Shruti, and thanks to all of our speakers.
Thank you for those excellent presentations. We're going to break for lunch. Please be back at 1:45. All right, everybody, if you could take your seats, we're going to get started with session 13. Phytoplankton and HABs, part 1. And take it away, Ted.
All right, everybody. Thank you so much hope everybody had a great lunch. We've got some really exciting sessions here about phytoplankton and harmful algae. Probably see a lot of folks you didn't see this morning who were at the CHABS workshop upstairs, and we've all trickled down here. So our first talk today in our session is going to be by Ellen Priest from the Department of Water Resources, and she's going to be speaking about harmful algal toxins in shellfish in the San Francisco estuary.
So well, they pulled this up. The talk I'm going to give is it's the first of a 2-year study where we are looking at harmful algal toxins along the salinity gradient. Oh, don't touch it. So this is kind of some preliminary or initial data that I'll be going through. And we'll have 1 more year of data collection that will be able to add to this. But this is a project that's funded by NOAA's Monitoring and Event Response Program, and then DWR is also providing funds to support it. So first, some background. I'm going to be talking about 2 toxins, microcystins and domoic acid. Microcystins are produced by a variety of cyanobacteria, and they're very stable. They can survive through a lot of different conditions, and they target the liver in human and animal bodies. And I have this cute picture of a sea otter up here because in 2010, 21 federally threatened sea otters in the Monterey Bay died from feeding on microcystin contaminated shellfish. And that really kind of brought awareness to this issue that shellfish can accumulate microcystins and be a potential risk to consumers that feed on them. Since then, a growing number of studies have found that a wide variety of different shellfish species can accumulate microcystins. As far as microcystins in San Francisco estuary shellfish, there's been some study in the Bay, mostly out of UC Santa Cruz. The first study looked at how wild marine mussels tested positive for microcystins during every month of the year. This was in the western portion of the San Francisco estuary as well as the Pacific coast. Another study out of the same lab placed caged mussels across the San Francisco estuary, including 1 or 2 locations just within the most kind of western boundary of the Delta. And they found that mussels tested positive for microcystins 82 to 100% of the time that the shellfish cages were deployed. And then the other relevant study is Pitamacorbula. So those are the really teeny little clams that have taken over the more saline portion of the bay, tested positive for microcystins from 2009 to 2018, with seasonally high peaks, seasonally being during the summer months when cyanotoxins are usually most prevalent in the system. In the Delta, I'm showing a figure from a recent study that myself and my co-authors published, and we looked at Asian clams, Corbicula, so the kind that are covering the Delta, the more freshwater clam. We looked at 10 sites across the Delta for 2 years, from 2020 to 2022, and I'm just showing one of the figures from that study. And I'm showing you this, that microcystins were present in all months of the year, although generally at very low concentrations in the Asian clams. And we also had these seasonally high peaks. So we had the most microcystin in July and August. And then there's 2 horizontal lines on this figure. The bottom one is the California threshold for safe fish consumption. And then the dotted one is for the World Health Organization tolerable daily intake. And you can see that we had a few exceedances of those thresholds during the summer months, but generally the toxins were below that. And in that same study, we also opportunistically collected crayfish and found toxins present as in the crayfish as well. So the other toxin that our study addressed is domoic acid. This is a neurotoxin and it's produced by the marine diatom Pseudonychia. Human and wildlife acute exposure to domoic acid presents as amnesic shellfish poisoning. It's of concern when domoic acid is at 20 parts per million, so quite high. And I'm showing a figure here that shows solid phase absorption toxin trackers. So these are passive samplers that are measuring toxins. This is the same study or same authors out of the University of California, Santa Cruz lab. And I highlighted in red Sassoon Bay because that's the closest to the Delta. And what you can see here is that there were low concentrations of domoic acid detected across the bay, including along the salinity gradient where our study takes place. So that leads to the knowledge gaps that we wanted to address in our study. First, are microcystins from the Delta transported downstream to San Francisco Bay? Second, is domoic acid from the bay contaminating wild biota in fresher portions of the estuary? And then third, is it necessary to conduct routine HAB toxin monitoring in that transition zone between the Delta and the bay? So for the talk today, I have 3 objectives. First, determine if microcystins accumulate in shellfish along the salinity gradient. 2, determine if domoic acid accumulates in shellfish along the salinity gradient. And then third, determine how long to deploy these passive samplers, the SPATs, to measure potential toxin transport. So we had 4 sites for our study. So going from west to east, we have Cal State Maritime, Martinez, the McAvoy Yacht Club, and then Pittsburgh. And then the pictures underneath this map show what the field sites look like at each of those 4 locations, and they match up going west to east. And it's a little bit hard to tell, but the 2 western sites are more open water bodies, whereas the 2 eastern sites are within marinas. For our methods, we collected crab, shrimp, and crayfish, and the USGS and Restore the Delta have really done a majority of the field work for this study, as well as students at Cal State Maritime. So collect for 2 times a month during June to October. So that's the summer period when we expect to have the peak microcystins, and then once a month in the other months of November for May. I mentioned it's a 2-year study. We've conducted the first year of that. The cages are deployed from 24 to 48 hours, and they're baited with hot dogs, which shellfish love. And then for the lab, we send the samples to UC Santa Cruz, who are doing the analysis for the toxins. The crab and crayfish, the shells are removed from them, so it's just the tissue meat, and the shrimp are analyzed whole. And we're using LCMS to measure the toxins. For the SPATs, these are deployed at the same time as the shellfish traps. And we are deploying them for 24 to 48 hours. So we deploy it at the same time we deploy the shellfish trap, and then they pick it up again when they retrieve the shellfish trap. And then we also have a longer deployment. So during the summer months, it's for 2 weeks. And then in the winter months, it's monthly. And these are also analyzed for via LCMS for microcystins and domoic acid. So first objective is microcystins and shellfish across the salinity gradient. These are the 4 sites that I mentioned. And so Pittsburgh is the top, the far eastern site is the top left. And then Cal State Maritime is our furthest west site, and that's on the bottom right. And so there's a few things that I want you to take away from this figure. So first, on the y-axis is the microcystins in nanograms per gram. And then on the x-axis, we have by date. The color reflects what species. So purple crab, the blue-green is crayfish, and the yellowish-green is shrimp. And then I have those same thresholds that I mentioned earlier. The California threshold for seafood consumption is the solid line, and the World Health Organization tolerable daily intake is the dotted line. You'll notice at the Martinez site and the Cal State Maritime site, there's not nearly as many points on the figures, and we've been less successful with capturing shellfish at these locations. But the two eastern sites do show exceedances of these health guidelines during the summer months. By both crab and crayfish, we haven't had any high hits in the shrimp yet. The second objective was domoic acid in shellfish across the salinity gradient. So this is the same setup that I just showed you for the microcystins with the far eastern site at the top left and the most western site in the bottom right. So this is domoic acid in nanograms per gram, and I mentioned the cause for concern is 20 parts per million. So we're well below that. But what we do observe is that we're having domoic acid hits, low concentrations of domoic acid in almost all of our shellfish. And so it's these same shellfish are contaminated with both microcystin and domoic acid in many cases. And one of our co-authors, Rafe Kudela, studies a lot of domoic acid, has been investigating how these low levels of domoic acid, kind of this persistent low level of domoic acid, may be of concern to biota in the system, even if it's not a human health concern. And so further investigation is needed on that. It's also interesting to us that we're seeing domoic acid kind of along the salinity gradient because it's produced by a marine diatom that we don't observe in this area. And so the toxins appear to be transported to these locations. And then moving on to the third objective was HAB toxins and spats across the salinity gradient. So first I'm going to talk about microcystins. So in this case, each individual plot is a month. So January at the top and then ending in December. And on the x-axis, I have the 4 sampling sites from east to west, and then the toxin concentration on the y-axis. And then the purpley color is a 1-month deployment. The greenish blue is a 2-week deployment, and that yellowish green is the short or 24 to 48 hour deployment. And what you can see is when we have the highest toxin concentrations, really June through October, we have pretty good agreement between the long deployed spats and the short deployed spats. And then we're not picking up a lot of toxin in the winter months. I took all of those locations and just plotted it together so we could see, taking away site, but how they matched up. And what you can see is in the summer months, once again, we have pretty good agreement between the short and the long deployment of these spats, with the short deployment on the X and the longer deployments on the Y-axis. Moving on to domoic acid now. So the same setup that we have these plots by month, January through December, but this tells a different story than the microcystin. So once again, those light green dots are the short deployments, 24 to 48 hours. And what you're seeing is that those short deployed spats are much more likely to get domoic acid detections than the longer deployed spats. Looking at May through August, almost all the long deployment spats are non-detects. And so that's an interesting finding to us, that these short deployments are more likely to detect the toxin. And I did the same thing where I plotted all of those points on one plot, and you can see there's a really poor relationship in detecting domoic acid between the short and the long deployed spats. So in conclusion, for the first objective, we show widespread microcystin contamination of shellfish across the salinity gradient. And at the far eastern sites where we have the most samples, microcystins exceed human health thresholds in summer months at the 2 eastern sites. We're trying to figure out some ways to have better luck capturing the organisms at the western sites, because microcystin is present in the spat samplers, which suggests that it very well could be present in the shellfish too if we could get more samples. As far as domoic acid, we have this widespread low contamination of domoic acid in all of our shellfish across the salinity gradient. And the detections are low, but they're persistent across time and space. And then for the spats, in the summer months, long and short deployments appear to have similar microcystin detection rates, but that's different for domoic acid. Short spat deployments are more likely to detect domoic acid than the longer deployments. I just want to thank everyone that's helped with this work, including colleagues at DWR, and then Alex Parker and Ali Johansson at Cal Maritime, and then University of California, Santa Cruz for processing and running all of these samples, as well as the funding. And that's all. I'll take any questions.
Okay, great. We have time for a few questions.
Yes.
Do you have any hypotheses for why the shorter spat deployments were more likely to detect the domoic acid?
Not yet. I just made those figures yesterday, so I don't know. We have to think through it a little bit more, and we'll obviously set them out for the next year or two and see if we're still seeing that same pattern. But it must be degrading off the resin, it would be my guess.
Thanks.
Thank you. Oh, Ted has a question.
I was, in the initial slides about microcystin in the clams and that you were seeing it still in the wintertime months. Some of the, essentially clams kind of acting like little living spats. Do you have a sense for the desorption or like the half-life of microcystin in a clam? Because it's like, it's probably pretty unlikely that microcystin's being actively produced in December and January, but it's still there present in the clams.
We did a decay rate calculation, and it was a couple weeks is what we were estimating for our clams. I don't know why. My guess would be that there may be toxins in the sediment. That's something we're investigating right now, is persistence of microcystin in the sediments at different locations across the Delta. Thank you. Oh, sorry. Who has it?
Sorry, I have.
Oh, yeah.
I was just wondering if you know of any mechanisms that degrade either domoic acid or these microcystins in the environment.
Well, microcystin degrades with sunlight. It's the number one thing that it can survive for or exist for long periods of time in dark places. And we've done cores of it and shown that it's pretty deep down in the sediment, suggesting that it may be down there for a while. And then I'm less familiar with what may be causing domoic acid. I don't know, Keith, if you have no, I don't.
Thanks again.
Okay, thanks.
Okay, so our next speaker is Keith Gregson from the USGS. Is going to be talking about genome resolved metagenomics on cyanobacteria.
Okay. Hi, everybody. So my name is Keith Gregson. I'm at the USGS, and I'll be talking with you about this genomic data set that we've been building up on cyanobacteria. But I really want to highlight Andrea Kust, who's one of the co-authors. She's the postdoc who worked on this project. She pretty much did all the work on it. Her partner works for the Nature Conservancy as a forest researcher in Africa. So she's actually living in Gabon, Africa, finishing up the data analysis, which is why I'm giving the presentation, not her. But she's really the one who's done all the field work and has been leading the charge analyzing these data. I also do want to highlight all the field staff that are out collecting and processing samples that make presentations like the ones I get to give possible. It's really IEP couldn't be done without you. So thank you. Whatever agency or group you're part of or private sector, it's an amazing amount of data that you give folks like me the opportunity to comb through and investigate. And the last acknowledgement, the USGS, we get funding from numerous different entities. So I want to acknowledge and thank all the different groups that have funded our various projects over the years. This project in particular was made possible by a Delta Science Program postdoctoral fellowship, which was awarded to Andrea. Andrea was a postdoc at UC Berkeley, so I also want to acknowledge her advisors and mentors there, Spencer Diamond and Jill Banfield. And we also partnered with Environmental Monitoring Program and Ted Flynn. So Ted and your group, thanks for giving Andrea space on the Sentinel. And she definitely wanted me to make sure she thanks all the folks she worked with when she was out there. So anyone in the EMP program that worked with Andrea and helped her out there in the field, we really appreciate your contributions to making this data set and project possible. So we're here to talk about cyanobacteria though. So cyanobacteria back in 1999 was the first report of a cyanobacterial harmful algal bloom in the Delta. It was determined that Microcystis was the bloom forming organism. This basically kicked off what will now be our 26th year of CHAB monitoring and research in the Delta. And we've learned a lot since, in these last 25 years. One of the reasons we care about cyanobacteria is because they produce these molecules called cyanotoxins, which are harmful to humans and animals. Cyanotoxins are classified as secondary metabolites, which means they're not used by the cell for just the normal growth, development, and reproduction. And so what that means is that you can have some cyanobacterial species and strains that produce the toxins and some that don't produce the toxins. There's numerous different types of classes of these molecules. I've listed here in this table the most commonly monitored toxins kind of globally in different monitoring research programs. And in the Delta, microcystins are the most commonly detected and at the highest concentrations. However, this figure I'm showing you here is an often shown figure from Peggy Lehman's 2021 publication. And it was the first time in one of her data sets that she detected anatoxins or saxitoxins. And so since then, we have had these somewhat consistent detections of these other molecules, albeit at low concentrations. And so I highlighted in green here all the different toxins that we detected in our data set as part of this study. So we basically detected all these common toxins except saxitoxins. And so now, since 1999, when that first research began and things were pretty focused on Microcystis and microcystin, what we know in the Delta is that we have multiple different cyanobacterial genera. There's Microcystis, there's a Phanosomenon, there's Delicospermum, there's Picocyanobacterium like Cyanobium. We also know that there's multiple cyanotoxins in the Delta, as you just saw. But one of the data gaps we have is we still have pretty limited information about the toxic compounds that are produced by these different taxa, and some of the differences between the ecology and physiology of these different cyanobacterial genera and species in the Delta. So our project objectives were to assemble the whole genomes of cyanobacteria in the Delta, and then from these assembled genomes, assess the diversity within the cyanobacterial genera, and then identify and characterize different gene clusters in these genomes that produce known toxins or other potentially bioactive metabolites. And then also look for genes used in nutrient cycling to characterize the diversity of nutrient utilization potential among the genomes. And so the main method we were using to do this is an approach called genome resolved metagenomics. So I just want to talk through that in case some of you aren't familiar with this. So in the top there, we have a picture of a cyanohab which has numerous different types of species of bacteria. And each bacterial cell has a genome that you can see in the circle there. So what we do is we collect a sample, put on a filter, sequence all that DNA, and you get all these short reads of DNA. So each about less than 200 base pairs of nucleotides long. But what you can do is you can assemble these short reads by looking for overlapping sequences that are the same. So when you get an overlap, you can kind of just extend out your read into longer sequences. And so eventually you can get sequences that are 10,000 to 100,000 base pairs long. And from those, you can then combine them together to actually form the genomes, which wind up being multiple millions of base pairs long. So now once you have the whole genome assembled, you can then look at the different genes that are present in that genome, look at the diversity of those genes, those functions, and really look at things in a whole genomic context. And so this approach differs a little bit from one of the common approaches used in the Delta called amplicon metabarcoding. In that approach, you look at a specific barcode or segment of the genome, and then you sequence all of that, and then you're able to analyze those data within the community, but you don't have that whole genomic context. You just have that short little section in each genome. And so the amplicon metabarcoding and the genome resolved metagenomics are very complementary methods. The metabarcoding is cheaper and faster, so it's easier to deploy. And so we're very excited because there's been numerous amplicon metabarcoding studies. This is one of the first genome resolved metagenomic studies. And so we're excited about the way these data sets will complement each other. And so we used both the combination of Illumina short read sequencing and PacBio long read sequencing, and then we assigned our taxonomy with the genome taxonomy database. So to collect our samples, as I mentioned before, we were primarily collecting samples at EMP sites. So we sampled 10 EMP sites from April to November in 2023, and we were able to supplement that with 11 USGS sites that we just sampled in July and October of 2023. And the samples were collected with a plankton net towed behind a boat. The net tows were 60 seconds, and we used two different mesh sizes, a 63 and a 20 micron mesh size to try and target different colony sizes and cell sizes. And then those, the material we collected in the net was combined into a jar, shaken up, and then subsamples were taken for cyanotoxins, DNA, and microscopy. And this wound up being a total of 104 samples collected in 2023. And if any of you remember 2023, it was a wet, rainy year. So it actually wasn't the best year to do a cyanobacterial research study. So we're very happy that we already knew we were going to be concentrating with these plankton nets because it was really important. We had to collect a lot of biomass. And so this is really more study about looking at the potential and diversity and the detections of what's in our system, and less about capturing like a really intense bloom and understanding what was going on in that bloom. So looking at the cyanobacterial diversity, we were able to assemble a total of 215 cyanobacterial genomes. These are the different taxonomy that was assigned to them. You can see that most of the taxa was from Microcystis. We had 79 genomes. In Delicospermum, we had 75, and those were the two main ones we were targeting. We were pleasantly surprised with this Koinonema. I didn't actually really know what it was, but these are a benthic taxa. So we often see like little benthic mats that have floated up to the surface. We must have caught those in our nets. And there's been a lot of questions just trying to understand and wondering about what the contribution of benthic cyanobacteria might be to food webs or toxins. So we now have a number of those genomes. And we also were able to recover some picocyanobacterial genomes as well. And these are the very small organisms that are 2 microns or smaller in diameter. There's a lot of those in the delta. And similarly, there's been a lot of speculation and wondering about what they might be, their ecological roles might be. And so now we have some genomes to assess that. So we're going to dive a little bit into this plot, which is kind of a lot, so bear with me. This is an average nucleotide identity figure. And so average nucleotide identity is a common method used for comparing genomes. You basically line up all your DNA sequences and just count all the identical nucleotides and get a percentage of identity. And so in this case, and generally, if it's above 95%, it's considered the same species. And so here, the bright yellows are almost 100% identity, so very similar genomes, and it's really just our darkest blue that might suggest a different species. And then these are pairwise comparisons, so the genomes are the same on the rows and the columns, and then they're all just clustered by their similarity. That's why you get these nice squares of color. So what we see is that and there's those 2 large yellow squares. Those are both a part of the Microcystis paniformis species, but we have 2 strains within that species, which is sort of corroborates a lot of the other work that like Ellen and others have been doing, that amplicon metabarcoding, which is showing that we do have multiple strains of Microcystis in the delta. Now we've got a little more genomic context for them. In the top left corner is a different species, Microcystis viridis, which is different than those other ones. And then you can see that diagonal of just yellow squares, those are all genomes that didn't really have as high of a sequence identity with other species. So those are kind of just like more diverse and just lower abundance taxa. I also want to highlight the genome taxonomy database. It's a microbial database that doesn't really use the same nomenclature as traditional cyanobacterial taxonomy. So that paniformis is effectively Microcystis aeruginosa, which is what we're usually used to calling it in the delta. For Aphanizomenon and Delicospermum, we kind of lump that together into what's called the ADA clade, and that's because there's a lot more uncertainty about this specific speciation within these two organisms. But Aphanizomenon is also very common in the delta. It makes these grass cutting like colonies in the delta. But unlike the Microcystis here, we kind of just see this big smear of yellow. So whereas we had a lot of different strain level diversity with Microcystis, it looks like we really just have primarily like one strain of Aphanizomenon in the delta. Delicospermum looks a little more like Microcystis, where we have that cluster in the top left of Delicospermum cercinale, and then this like smattering of rarer and less abundant taxa that don't form these big clusters. So one of the things we're then interested in and what we can do with these genomes is look at what is the biosynthetic gene diversity. What are these genomes and organisms capable of producing? So this figure is, again, dense. So what we've got in it, on the bottom axis is the different genomes, and on the y-axis are all different types of bioactive compounds that could be produced by these genomes. And so every time there's a circle, it means that that genome, we detected those genes for that potential compound in that genome. And what you see, we're just going to key in on a couple of them, but in the middle, is there a laser point? No, it wasn't working for me. Can you just click that logo? Yeah, it's not coming up, right? Sorry. Okay, that's right. All right. Microcystin is in the middle there. And what you see is that we only recovered microcystin genes from the Microcystis genome. And so those Aphanizomenon and Delicospermum genomes do not seem to have the genes for microcystin. And there had been speculation that Aphanizomenon and Delicospermum were not microcystin producers, but this is some of the first evidence we have that they really might not be producers. So that was an exciting finding. However, geosmin is down there a little bit lower than microcystin. We do see geosmin genes for Aphanizomenon and Delicospermum. And geosmin is a taste and odor compound producer that affects drinking water treatment. And so if we have elevated Aphanizomenon or Delicospermum populations, that might start to have larger negative impacts for drinking water treatment. Additionally, some of our picocyanobacteria like Cyanobium, which is the third one over, and our Arsenicocollis don't have many gene clusters. So it seems like the picocyanobacteria are probably not producing that many harmful compounds in the system. But that is in contrast. If you look at Microcystis, how many dots there are, like some of these cyanobacterial taxa can produce a lot of compounds, and we don't really even fully understand what all these compounds do because they haven't all been studied thoroughly yet. And then the last point to make here is what disappointing finding is we never recovered any anatoxin or cylindrospermopsin genes in our data set. So I was really hoping that we might be able to get a sense of what organisms are producing that, but that really remains a mystery because we didn't recover those genes. So taking a deeper dive into the Microcystis genomes, what I did here was I took that same ANI plot and I am showing with the red arrows which genomes did not have the microcystin gene cluster, and then the numbers show which ones did. And so what we see is that large Paniformis or Aeruginosa cluster, those two different strains, almost all of them have the microcystin gene. So that is probably a very likely toxigenic taxa. In contrast, the Viridis cluster, which is in that top corner, none of those genomes had the microcystin gene cluster. So that is probably a non-toxigenic taxa. And then for those singleton genomes, only 6 of the 17 had the genes for microcystin production. So it seems like we have sort of like low diversity of strains that can produce the toxin, and then this higher diversity of non-toxic genera in our data set. The benefit, okay, almost done. We don't only have to look at toxin genes, we can look at other ecologically relevant genes. So we also took a look at some of the nitrogen and phosphorus cycling genes. So here what we're looking at is along the x-axis are different genes for nitrogen cycling or uptake, as well as phosphorus. And we see a lot of similarity in the nitrogen genes. The purple is nitrogen fixation. And what we see is that our ADA clade, our Delicospermum and Aphanizomenon, as well as the Piscivorous Vermopsis, can fix nitrogen, whereas the Microcystis cannot. So that's sort of a known trait difference between these taxa. And then we also see for the phosphonate, the orange ones and the pink on the right side, those are genes for scavenging organic phosphorus, And we don't see a lot of genes for that. And that actually makes a lot of sense because scavenging organic phosphorus is only useful if your inorganic phosphorus becomes limiting. And we have a lot of inorganic phosphorus in the delta. So it's probably not that advantageous to be able to have genes for acquiring organic phosphorus because there's so much inorganic to go around. So our next steps are, one of the things we want to start to look at is determine if some of the reads from the other amplicon-based studies can be mapped and aligned with our genomes. We can start to link our strains that we've found in our data set with some of the other molecular work and data sets that have been developed recently. We want to further investigate the nitrogen, phosphorus, and other ecologically relevant genes and look for pathways and differences between the genomes. And then also compare the environmental data in our data set with the different genomes and genes to look at how these environmental conditions correlate with the gene and genomes. And Vivian Klotz had a poster during the poster session already starting to look at this. So we're really grateful for Vivian's contribution to this project. So there's some references. And with that, if there's time, I will take any questions. And thank you all for attending my talk.
We have time for questions.
Rosie?
So if you weren't seeing the genes for slender, two of the toxins, but you were seeing them in the environment, what do you think is going on?
I anticipate that. It's definitely a mystery. It does happen. So it could, there's kind of two hypotheses. One is that really rare organisms, so maybe the toxins were dissolved or kind of or loosely absorbed particles. So the toxin was there, but the toxin producing organism was there, or it was just at a very low abundance that we couldn't kind of assemble it within the mishmash of all the DNA that we were working with. But that's kind of the best I've got right now. And as I mentioned, our abundances were pretty low because it was that 2023 year. So maybe in bloomier years or in sampling different types, we might eventually figure it out.
Can you just comment on how often we have this data coincident with cyanotoxin data? Because just kind of building on Rosemary's, like we don't know if the cyanotoxins were present actually in those water samples, do we? Or we would do that data?
So yeah, so everything was co-collected. So we were subsampling and then analyzed the toxin sample. So so I guess that's another thing if the subsampling splits weren't perfect. But yeah, we measured every sample for toxins and then did all the DNA extractions and genome assemblies.
Will you look at mRNA in the future?
Yeah, so actually we do have some transcriptomic samples that were run for transcriptomics. So we haven't looked at that data too much, but that would kind of be the next step to actually look at like what genes are being expressed in the different samples and under different conditions.
Of the potential of the strains, the toxic strains, the ones that had the MCY gene, did you detect any that had partial?
Partial?
Yeah.
Yeah, so I didn't mention, we did, I'd say a thorough initial look and most of them looked pretty complete. So I think almost all of them, the majority of them are actual full sequences. Yeah, they looked very complete and similar like synteny and organization as other reference sequences. All right, thank you, everybody.
Okay, so our next speaker is Sylvia Angles from DWR, and she's going to be talking about some DNA metabarcoding on cyanobacteria and phytoplankton.
It's working. Yeah, hello everyone. My name is Sylvia Angles. I'm with the DWR. And well, I'm gonna talk about a project that we're working on. I'm gonna show the first results actually. And this project is exploring the use of DNA metabarcoding for cyanobacteria and phytoplankton monitoring in the Delta. So Keith made some things, set the ground for me a little bit. So that's helpful. Thank you. But first, I would like to acknowledge the other collaborators of the presentation: Andrea Kust from UC Berkeley, Alexa Camilleri Evans, Ted Flynn, and Daphne Gill from DWR. Also, the whole crew of and the environmental monitoring program for facilitating the fieldwork, and also the genetic monitoring lab at DWR for the huge help in all the lab aspects of the project. So why do we want to monitor phytoplankton and cyanobacteria? Well, phytoplankton play very important ecological roles in aquatic systems, from the production of oxygen to the removal and transfer of carbon, transfer through the food web, nutrient cycling. They also serve as food for other trophic levels in the food web. They are also very diverse. They are mainly composed by microeukaryotes and also cyanobacteria, which are mostly photosynthetic bacteria. So all these, we want to, due to this importance in the ecosystem, we normally monitor their abundance and their community composition. But phytoplankton and certain species can also produce harmful algal blooms, as we've been talking in this past two presentations. They can produce toxins and metabolites or high biomass that can cause harm to the ecosystem and to humans and animals. And well, in the Delta is not exception, unfortunately. There have been like cyanobacteria blooms observed in the, I should say, 25 years perhaps, or 30 years ago. And the main taxa found, the most common that produce blooms are Mycocystis, Aphanizomenon, and Dolichospermum, as we already seen. So the phytoplankton traditionally has been done using, the phytoplankton monitoring has been done using microscopy, which is a very well-established technique. It provides valuable information on abundance and carbon biomass of phytoplankton, but it's slow, it's time-consuming, and it requires high taxonomic expertise of the skills and expertise of the taxonomers. So somewhat there is a limited amount of samples that can be analyzed. And it also poses some challenges in terms of identification based on morphology, basically small taxa or taxa that don't preserve well with the preservation used. And sometimes it's difficult to achieve species level. So for this, there is a tool, the DNA metabarcoding, which has been like getting more perhaps importance lately in monitoring of phytoplankton and other organisms. It's a technique based on like Keith already explained, based on a short sequence of the DNA, like we could call it a DNA barcode. And you will normally choose a marker gene, a gene marker. So it's sensitive, it's accurate, it's scalable. We can do a lot of samples at the same time. It's cost efficient then. We can identify more than one group of organisms with the same sample. And it allows for the detection of this small taxa that we were talking about, that it's perhaps difficult with microscopy, with taxa that don't preserve well. So it can provide also a screen, increased resolution at the species level. But the problem is that it's semi-quantitative, that we say we don't really obtain, for now at least, an abundance, or we can not obtain carbon biomass values. So the questions for this project were, well, to explore the application of DNA metabarcoding to improve the phytoplankton and cyanobacteria monitoring that we are currently developing, well, carrying out. with the goal mainly to support management decisions. So the main questions were, what is the taxonomic diversity that we are able to detect with metabarcoding? What taxa are there? How many? How does metabarcoding data compare with or complement the data we are obtaining with microscopy? And specifically for cyanobacteria, which of these taxa that we are potentially detecting are potentially toxic? Are we able to detect them? So for that, I collected samples at 7 stations, 7 EMP stations in the Central Delta here in this area. I collected them monthly from July to November in 2023. Again, a wet year, but it's okay. Then also this past year, from May to November, again, another wet year. But for this presentation, I'm gonna focus mainly on the data, the samples of 2023. I collected grab samples from the surface. Also in 2024, I collected samples at 1 meter depth. And this sample, this grab sample was filtered to concentrate DNA. And I also took a sample for microscopy analysis. Additionally, I also analyzed using DNA metabarcoding the same samples that Keith showed from Andrea Kust, that these samples were collected with doing net tubes. So the same part of it, for part of the DNA extraction I used to do the metabarcoding part of cyanobacteria and also phytoplankton. So I'm going to present a little bit of as a comparison because we wanted to see how much well, how do the data that we are able to obtain compare from grab samples to net tubs. And as you can see, there's the concentration was pretty different from the net tubs to just the grab samples. This is a little bit the metabarcoding workflow. Everything was done in-house, which we are very happy, at the gem lab, mostly the wet lab part. And each step had like its own implementation and tuning. So we are happy because we've been able to obtain data all in-house. For this case, the results that I'm going to present, I use as gene marker the 6TNase ribosomal RNA gene for cyanobacteria, since they are bacteria. And then for the rest of the organisms of phytoplankton, I use the 18S, a region of the 18S ribosomal RNA gene. And then all this, the community composition obtained after the bioinformatics analysis, I've compare them with the microscope data obtained from the same samples. So here are the results. Let's start with cyanobacteria. These plots show the data obtained with metabarcoding of the grab surface samples. We obtain high diversity of taxa of cyanobacteria. And this plot here on the top right, these purple bars are these picocyanobacteria that Keith already mentioned as well. So they were dominant mostly. So I removed all these taxa to kind of see a little bit better the other cyanobacteria taxa, which is this plot here in the center. We have the stations in the y-axis, the relative abundance of reads and facet by months. And we can see again, like we detected a large number of species of taxa. I plotted at the genus level to simplify things. And we were able to detect the main common taxa, Aphanisomena, Dolichospermum, and Microcystis. And as you can see, that we also observed like a temporal dynamics. At the beginning of the sampling study in July, Dolichospermum was the genus more common and abundant, dominant, let's say. Then towards the end, it was a phenomenon, so we saw like a little bit of a transition. And then microcystis, we had more occasional detections. Again, it was a wet year, so there was not much microcystis, but they mostly occur throughout all the months, but no clear pattern, temporal at least. So these are the results of cyanobacteria composition in revealed by microscopy. We also detected that the most abundant was small size cyanobacteria, so I remove it as well. So we can compare better. And we see that we did kind of detect, well, the results detected less taxa cyanobacteria, but we did detect the main species, Panisomnon, Dolichospermum, and Microcystis. But that pattern, temporal pattern that we could observe with metabarcoding was not as clear with the microscopy data, apart from perhaps a Panisomnon at the end of the sampling period. A few detections of Dolichospermum and Microcystis here and there, but that was what we obtained. So I wanted to compare a little bit at the richness at the genus level between the metabarcoding and microscopy. So we can see here that the taxa detected was higher. The number of taxa was higher with metabarcoding, and we had like kind of small overlap between the two methods, but we did detect that, like I said, the three main common taxa and also Pseudonabaina. We did also detected other potentially toxic species with metabarcoding, so that's a positive thing. And here I plotted to compare all the metabarcoding of cyanobacteria with of the netto samples. Again, we didn't get a lot of the pico cyanobacteria because it gets filtered through the mesh 20. It's smaller than 20. So we with these samples we basically had the larger cyanobacteria concentrated. We detected again the same similar species taxa in general, a few more detections perhaps with the netto samples, but again the same taxa of the most common ones, and kind of like a similar temporal pattern, except that perhaps for microcystis, the detection of occurrences of microcystis was higher with the netto samples. So this is the green, the light green bars. Well, I think that's most probably because with the netto we would do like a larger spatial coverage. And in that case, that year, the colonies of microcystis were more sparse, so we were able to kind of concentrate and get perhaps improved representation of microcystis with the nettos. The other two taxa seem like similar results. So let's move now to phytoplankton other than cyanobacteria. These are the results obtained with metabarcoding. We obtain good data for all the classic phytoplankton groups, diatoms, the classic ones, diatoms, cryptophytes, chlorophytes, which are green algae. These were the main groups, the more dominant groups, chlorophytes, cryptophytes, and diatoms. And we kind of see also again like a temporal pattern. Perhaps there is like a decrease in diatoms towards the end of the study period and an increase of green algae towards the end of the study period. And then we saw like a slow, low, but kind of a detection of dinoflagellates during all the this dark green bar during all the study period. If we compare with the microscopic data, we obtained the same groups, which is good for metabar coding. And the most dominant ones were diatoms, also cryptophytes. But contrastingly, we had like the dominance of green algae was a lot lower than what we observed with metabar coding. I think that the reason might be because there were a lot of small taxa in the chlorophytes. It's something that I need to dig in a little bit more, but that seems to be the case. And then the detections of dinoflagellates were very occasional. We see them 3 times. And while with metabar coding data, we were able to detect better representation of dinoflagellates. So to put a little bit everything together, here's like 2-3 maps of the genus richness for phytoplankton without cyanobacteria detected with metabar coding on the top. And then the genus richness of detected by microscopy on the bottom map, 3 map. We see again that we detected the same groups. They have a little bit different names, but chlorophytes are green algae. But the number of genera, as you can see here, was a lot higher in a few of the groups, chlorophytes notably, dinoflagellates, chrysophytes, other groups that are perhaps, well, that is difficult morphologically to, based on microscopy, to identify, that we were able to recover them with metabar coding. So the main takeaways are metabar coding detected a higher number of taxa compared to microscopy, demonstrating it's capable. It's a good tool to capture cyanobacteria and phytoplankton biodiversity. There was limited overlap of cyanobacteria in cyanobacteria identification between metabar coding and microscopy, but the predominant taxa were detected by both methods. When we compare the surface grab samples with the nettoes, they both seem to capture the temporal shifts in cyanobacteria community composition, but microscopy did not fully capture that temporal dynamics that we saw. And finally, metabar coding and microscopy showed agreement in the main phytoplankton groups, but they revealed different proportions and temporal dynamics in the most common ones. And there's still a little more work to do. Stay tuned, but yeah, that's all I have. Thank you very much. We have time for questions.
I think we have time for maybe one question.
I'll be quick. So I'm imagining that different sizes of algae produce different amounts of genes, or
Yeah.
do you have any sort of handle on if that's kind of a cause for the differences?
Well, yeah, so there are different, depending on the species and the size, there are different copies of genes, depending a lot of the taxa and the group. So that could be a reason. Dinoflagellates are known for have a huge amount of copies. But what I was showing here in terms of genera was that different kinds of taxa. So that doesn't depend on the genes, the gene copies. So, but yeah, in terms of proportions, yes. That could be.
Stephanie has an online question.
Oh,
Yeah, I think it'll be quick. Sarah says, great talk. And how complete are the taxonomic species databases for these groups? Is it possible that some of the species ID'd by microscopy were not represented?
Yeah, definitely. Yeah, that's one of the reasons of the lack of overlap. Yeah, that's something that I would need to look a little bit better, and if they are represented or not on the GenBank, on the database and genetic database. But yeah.
Thanks again, Sylvia. Hey, and for the final speaker of this session, we have Alex Ryan for a comparative analysis of primary production and nutrient cycling in San Francisco Bay.
Hello, everyone. I'm Alex Ryan from the USGS Biogeochemistry Group at the California Water Science Center. Today, I'll be giving you a very quick update on a project that we've been working on in conjunction with the San Francisco Estuary Institute. We're looking at water quality variability in the shoal and channel regions of the San Francisco Bay. And while data was collected from both the South and Central Bay, noted here, this talk will only be discussing data collected in the Central Bay portion. And zooming in slightly, we can see the bathymetry of the region, clearly highlighting the channel with the darker hues illustrating depth. And the channel has been the most accessible part of the bay to collect data from. Historically, we've collected water quality data in the channel, but this is not without sacrifice as the channel isn't representative of the entire system. In fact, approximately 65% of the bay is classified as a shoal type of one form or another. And we have evidence that suggests that phytoplankton growth occurs more readily in the shoals. These shallower regions, because there are so many of them in the bay, we wanted to investigate the differences between shoals and channel regions and potentially identify any connections between the two. And we did this by looking at things like nutrient availability, environmental conditions, and hab formation, with the goal to hopefully characterize these differences. The data collected for this project involved high-resolution mapping survey data, as well as using a continuous flow-through system, as well as discrete sample collection at stations throughout the bay. And our high-res mapping is interesting because it collects data for a variety of depths, including the shallowest regions accessible by boat, as well as in the channel, allowing for cross-comparison of nutrient levels and phytoplankton growth. Although many parameters were collected, this talk, due to its short nature, will only focus on chlorophyll and nitrate concentrations. And for the statistical methods, we used two tests, the first of which being a mixed model ANOVA, which made comparisons between channel and shoal groupings over time. And the results indicated that all groups were statistically significantly different, which validated the use of a post hoc Tukey's HSD on a significant difference test, which compared shoal versus channel groupings over time for each mapping event. And the results are a little overwhelming at first, but bear with me. So time is read like a book from left to right, and we have, I don't know if this works. No. Well, so we have mapping events, which are each time that the surveys went out and collected data. And the colors are probably the first thing you're going to notice here. And the main thing they're meant to show is the Tukey's results, showing that between shoal and channel groupings, shown here on the bottom, the x-axis, that they're all different. Primarily, chlorophyll concentrations were elevated in the shallower shoal regions compared to the channel throughout the entire duration of the study, except for one mapping event here in September of 2022. And this anomaly where chlorophyll concentrations were lower in the shoals compared to the channel is, we attribute that to the infamous heterosigma bloom that impacted the bay from August to September of 2022. And following this bloom, we can see the typical trend of chlorophyll being elevated in the shoals compared to the channel for the remainder of the study. And here we have nitrate concentrations set up the same way. And there's an inverse relationship with nitrate compared to chlorophyll concentrations as those hungry phytoplankton use up nitrate in the shallower regions. So we see nitrate concentrations lower in the shoals compared to the channel, all except for, you guessed it, this time here in September, where nitrate is elevated in the shoals compared to the channel. And once again, that's attributed to the hab formation of the, Anyway, we have the channel and shoal regions are then returning back to their normal typical concentrations, with the shallower shoals having lower nitrate for the remainder of the study. And you can see that inverse relationship here as this is a 1 hour window time series of nitrate in orange, chlorophyll in green, and you can see that as the boat traversed through different shoal types, that inverse relationship is maintained despite the flip-flopping. And I want to leave you with 3 main points. The first being that the shoals are statistically significantly different from the channel based on multiple parameters, even though only 2 are talked about during this talk. And as well as shoals, the shallower regions facilitating phytoplankton growth at a higher level than the channel, which means that if we want to do accurate monitoring of this region, we need to include shoals in our studies. And I want to give a special thank you to USGS Priority Ecosystem Services for funding this project, as well as SFEI for making this work possible, and all the field crews at BGC for collecting this data.
All right, we have time for questions if you, yeah. Yeah, questions.
Hi, that was a great talk. Are you guys going to be incorporating any other kinds of data to look at the shoals since they're a little harder to get to?
I mean, besides the nitrate and chlorophyll concentrations, like looking at other data?
Yeah, like any other ways of getting it, getting data, collecting data?
I have no idea. I'm not sure.
All right, everybody. That was the end of our session. So everyone, last round of applause for all the speakers. Thanks so much.
Thanks, Ted. We're going to start our next break. Just a heads up, there are 2 water bottles at registration that were found in common areas. So if you're missing your water bottle, it's probably there. And then a reminder that posters are probably taken down now because they had to take the boards down. So if you left your poster upstairs, you might want to go claim it. It should be by the door somewhere. We will meet back at 3:15. Thanks. Okay.
Yeah.
All right, if everybody could take their seat, we're going to go ahead and get started with session 14, our final session, phytoplankton and HABs, part 2.
Okay. Okay. Hi, everyone. Congrats on making it to the end of IEP Day 3, the last session. My name is Hannah Cheney, and I am with the Delta Stewardship Council, and I will be the chair of this session, which is Phytoplankton and HABs Part 2. Before we begin, I do want to let you know that our last speaker, Francis Wilkerson, is unfortunately unable to attend today. So this session might be a little bit shorter than anticipated, but without further ado, I want to introduce our first speaker today, who is Skylar Nardelli, a postdoctoral biologist at the USGS California Water Science Center. Let me switch. One second, sorry. Yeah. All right, thanks you guys. Here you go, Skylar.
Hi, everyone. Thanks so much for hanging in there to the end. Today I'm going to talk about the massive Heterosigma Akashiwo harmful algal bloom that occurred in San Francisco Bay in August 2022 and the broader Protus community dynamics that help shape it. So, as you all well know, San Francisco Bay forms the lower portion of the San Francisco Estuary, and today we're really going to be focusing on the Central and South Bay. And when compared to other coastal estuaries worldwide, the San Francisco Bay is in the top 87th percentile for nitrogen loadings. Yet we do not see annual harmful algal bloom events like we do in other systems like the Chesapeake Bay or the Long Island Sound. And the working hypothesis is that light limitation from high turbidity combined with strong tidal mixing and grazing by benthic mollusks combine to produce a low productivity estuary despite the high nutrient concentrations. We do see blooms though on an annual basis. We see modest spring diatom blooms in roughly April and then sometimes a small fall bloom in roughly September. But in August 2022, we saw record high chlorophyll A concentrations in San Francisco Bay associated with a massive bloom of Heterosigma Akashiwo. And this led to widespread low dissolved oxygen and fish mortality throughout the bay. The bloom emerged in late July near Alameda and spread throughout the bay in August to fully encompass it in late August. And then it dissipated really rapidly by August 31st. And this bloom was really an indicator of changing conditions in the bay, which may lead to an increased potential for high biomass HAB events in the future. So what do we know about Heterosigma? It is a prominent fish-killing rhaphidophite. It's found worldwide, and it forms blooms that last weeks to months. It can withstand a broad range of salinities and temperatures and light conditions. And for context, our August 2022 salinities were kind of at the higher end of their salinity tolerance, and the temperatures were in the middle of their thermal tolerance, so nothing abnormal in 2022. They're very adaptable organisms, and they thrive in all types of environments. They exhibit high photosynthetic efficiency, rapid nutrient uptake, they're motile, mixotrophic, they can form cysts to overwinter, and they form mucus. And in the literature, there are other instances where we've seen Heterosigma blooms in the Northwest United States and British Columbia. Blooms often form in late spring, and they're driven by freshwater inputs that stratify the surface waters and also input high nutrients. However, there's other instances like in the Nervion River estuary in Spain where blooms occur in the late summer when discharge is at a minimum. And the August 2022 bloom matched the conditions found during the Nervion River estuary blooms where the actual environmental drivers are a lot less clear. So with not a ton of direct evidence from the environment as to the cause of the bloom, the goal of our study was to look at the bloom from a community perspective to see if the summer 2022 Protus community was anomalous. We also wanted to analyze and see if the changes in the community created opportunity for Heterosigma to bloom. And also we wanted to look at the community data in the context of the bloom decline. This map shows the stations where data was collected for the analysis. And I'm going to be showing figures today with data characterized as Central Bay and South Bay. And this is because the Central Bay has more exchange with coastal areas through the Golden Gate Bridge, whereas South Bay has longer residence times and was where we saw the worst of the heterosigma bloom. So at the triangle stations, they're located in the channel here. Data was collected 1 to 2 times per month off the RV Peterson, which is one of our larger research vessels. And then the circle stations, which are mostly confined to the shoals, data was collected roughly monthly and opportunistically off the RV Aiken, which is one of our smaller research vessels. The red stations are where samples were collected for metabarcoding of 18S ribosomal RNA genes. And for those of you who aren't familiar, the unique DNA sequences of 18S ribosomal RNA can be used for taxonomic classification of eukaryotes. And so we identified 27 broad protist groups, including phytoplankton and microzooplankton. And the relative abundance of these groups are portrayed in all the taxonomic figures in this presentation. So the 27 protist groups I mentioned can be seen in gray in this NMDS plot. Samples are color-coded by year, and the different shapes represent the different summer months. And the ellipses over top represent standard deviation. And you can see that the brown 2022 samples cluster separately from the other years. And this includes samples from months other than August, which is when we saw the heterosigma bloom. So there's something kind of bigger going on this year. To confirm this, we ran a cluster analysis. The color scale portrayed is an index of drought conditions on a scale from wet to critically dry. And in general, the years kind of cluster out by water type. But you can see that 2022 clusters separately from the other drier water years. So to investigate this, we looked at summer 2022 community succession patterns. And I'm not going to show all of the data today, but here are some of the key groups that contributed to differences throughout the summer. These plots show average relative abundance on the y-axis and month on the x-axis. And then the red lines represent data collected in the Central Bay, and the blue lines represent data collected in the South Bay. And because we're looking at the protist community, I just want to highlight that not all of these groups are autotrophs. So to start, in June, we saw a large diatom bloom in the South Bay. This figure on the left here is an anomalies plot. And so we're comparing summer 2022 to summer data collected in other years in our time series. And this top plot is looking at chlorophyll A. In the middle, we have suspended particulate matter. And the bottom plot shows silicate concentrations. And the blue bars are positive anomalies, red bars are negative anomalies. And what you can see in June is that this was an anomalously large bloom for June, and it was actually the largest June bloom in our entire time series starting in 1980. And it corresponded to a lot of suspended particulate matter in the surface water and large drawdowns of silicate. And this bloom appeared to have originated in the lower South Bay, which is often characterized by high productivity because it receives discharge from one of the largest wastewater treatment plants in San Francisco Bay. And it has a kind of physical feature that helps contain the water, creating longer residence times. In July, we saw a community representative of conditions following a large diatom bloom. There was a high relative abundance of heterotrophs that likely took advantage of the high suspended particulate matter as a food source. In particular, the Central Bay was dominated by the species Ebria tripartita, which is a marine flagellate that feeds on chain-forming diatoms like Skeletonema and Thalassiocyra, which were readily abundant during the June diatom bloom. We also saw a low composition of dinoflagellates. On average, there were less than 1% in early July. And this is abnormal. We usually see around 10 to 15% in early July. So this is really an indication that either the high prevalence of heterotrophs were out competing or consuming the dinoflagellates. And we also see a small, an autotrophic community that's dominated by small cells, which is typical following a large bloom because small cells have a higher surface area to volume ratio, so they can better uptake nutrients in low nutrient conditions. And they also are better at staying up higher in the water column. They don't sink out as readily. So based on these June and July community dynamics, there are two potential pathways operating in conjunction with other bottom-up drivers that could have led to the heterosigma bloom. So the first involves bottom-up influences. The June diatom bloom significantly drew down silicate concentrations, which likely limited further diatom growth in July and provided an opportunity for other taxa to increase. There was also high suspended particulate matter, likely phytodetritus, and this reduced the light availability in the surface waters. And so this selects for more heterotrophic and mixotrophic species that can utilize other energy sources other than photosynthesis to acquire their energy. And a lot of these heterotrophic and mixotrophic species are also motile, so they can move around to acquire nutrients more readily. So it's possible that heterosigma may have been able to thrive in these conditions and outcompete other heterotrophic and mixotrophic species that were present, especially since heterosigma has been found to be toxic to some protus graziers, such as large ciliates. The second pathway involves top-down influences. So, for example, the dinoflagellate gyrodinium is a known predator of heterosigma, and Ebria tripartita, although preferring chain diatoms, have been observed to eat dinoflagellates. So it's possible that heterotrophic species like Ebria may have exerted grazing pressure on predators of heterosigma, which in turn would release that grazing pressure on heterosigma and allow them to proliferate. So likely a confluence of these top-down and bottom-up community dynamics led to the heterosigma bloom that we saw in August. And this bloom reached average chlorophyll A concentrations of 137 micrograms per liter on August 25th. You can see how red the water got from this massive bloom. And then it crashed by August 31st, where we saw an average relative abundance of only 1%. So timed with the heterosigma bloom collapse, dinoflagellates increased dramatically, and this was really dominated by the genus Gyrodinium. And since Gyrodinium are predators of heterosigma, there's a possibility that they may have contributed to the heterosigma bloom decline. And some back of the envelope calculations suggest that they may have accounted for up to 20% of the decline. And, alternatively, they just may have thrived in these post-bloom conditions. Other hypotheses for the decline of the bloom include depleted nutrients, viruses, which we haven't been able to look into much, and other competition. There was also an increase of mixotrophic species following the dinoflagellate increase and other small cells, which, like I mentioned before, are characteristic following bloom conditions. So in conclusion, we looked at the bloom from a community perspective, and we found an uncharacteristically large June diatom bloom that led to a largely heterotrophic and small-celled community in July, with very few dinoflagellates. The absence of dinoflagellates, which could include predators of heterosigma, plus environmental conditions that select for mixotrophs, such as high detritus, low silicate concentrations, may have provided an opportunity for heterosigma to proliferate in August. And then gyrodinium increased after the collapse of the bloom, which may have helped contribute to the collapse, or they might just thrive in these post-bloom conditions. So if you take away nothing else from this talk, I hope you understand the value of looking at events like this from a community perspective. Not all blooms are driven exclusively by bottom-up controls, and understanding the ecology of the blooming species and its relationships to the broader protus community may help researchers and managers predict the occurrence and magnitude of future harmful algal bloom events. So with that, I'd like to thank all the field and lab techs that helped collect this data, as well as Ben Genetics for sequencing the 18S data. We received funding from the USGS Water Mission Area, as well as emergency rapid response funding from NOAA to support sampling during the bloom. And then USGS folks from my group are also co-PIs on a NOAA MERHAB project to advance monitoring technologies in the San Francisco Bay, which has helped support these analyses and the development of molecular data for HAB monitoring. And yeah, with that, I'll take any questions.
Yeah, I'd say we have about time for like 1 to 2 questions, if anyone has them. We'll take them from the room first. Anyone online? Thank you so much. I guess we're a little bit ahead of time now, but I want to introduce our second speaker, Sienna White with UC Berkeley. She's a third-year postdoctorate student, and yeah, sorry, you can come on up now.
I'm just a grad student.
Oh, okay. Okay. Thank you.
Hi, everyone. Great to see you today. My name is Sienna White. I'm actually a PhD student. Sadly, don't have my PhD yet. But today I'm going to be talking about vertical mixing in dead-end channels of the Delta. And I just want to acknowledge my co-authors. These were amazing collaborators to work with, and they contributed tremendously to this project. But this will be a little bit of a different talk. I come from an environmental fluids background, and I'm really interested in sort of the physical characteristics that make up the Delta, specifically the South Delta, in the hopes of really sort of like trying to zoom in on some of these environmental drivers that cause bloom events to occur. And so kind of broadly speaking, we all know that the Delta's experienced more and more harmful algal blooms in the last couple of decades. And I've highlighted here a couple kind of typical regions that we think of as HABs hotspots. And specifically in the South Delta, you'll hear people talking a lot about these dead-end channels, such as the Stockton Shipping Channel. And this is this idea that there's a lot of these regions in the South Delta that are pretty stagnant. You know, it's like a channel terminus, and they have long residence times. And again, we don't really get out there much. We don't monitor them, but there's a lot of thought recently that they might be sort of the places that these bloom events start, and then maybe the bloom advects outwards. So we're interested in the dynamics in these dead-end channels and really starting to zoom in on how that might affect different species of phytoplankton. And so, again, really broadly speaking, this is from a review from Kudela et al. But in general, scientists tend to agree that in the Delta, microcystis are benefited when they have high nutrient availability, and then they have low turbulence, such as long residence times. And then warm temperature and high light availability are also really good for starting a bloom. And then, of course, the converse would be bad for microcystis. And the thing I really want to touch on here is that it's a little complicated from a physics perspective to differentiate between low turbulence and long residence times. It's really possible that these two things can overlap. And so when we think about driving these big statistical models where we want to talk about residence time and low turbulence levels, you have to be pretty precise in how you define these things. And so I want to make sure we're on the same page. So I'll just define a couple broad terms. The first is turbulent diffusivity, which I'll be talking a lot today. This is an estimate of the rate at which a turbulent process can mix a gradient. And so the key thing I want to note here is this isn't a real quantity like temperature. This is a parameterization. So this is, again, sort of an average of you have some turbulent process, and you're saying, in general, if I have my pink water up here and my blue water down here, higher turbulent diffusivity is going to mix that out into purple water faster. And then, of course, we have the photic zone, which is really important because this is the depth at which 90% of incident light is extinguished. And that really matters if you're a phytoplankton, because your whole life relies on you getting into the photic zone in order to reproduce and grow. And in the Delta, this tends to be on the order of centimeters to meters, depending on how much turbidity is in the water column. And so I want to note a really important physiological difference between two main species of phytoplankton that I'll talk about today. One is that diatoms are heavy and they can't swim, and instead they sink very slowly, compared to microcystis, which have the ability to swim very barely on the order of like a little bit of like centimeters per hour. And so the idea here is that these slight differences in their motility might make a big difference depending on the currents in the water column and the turbulent mixing. And mostly what I'm trying to say is that without turbulent mixing, if you're a diatom and you're just sinking all the time, you're never going to make it into the photic zone. So you're relying on vertical mixing to get up and grow. And then, of course, that if you're in a region that's really stagnant and has a low level of turbulence, if you're a diatom, you're not going to be able to get in the photic zone, but also that region would have a high residence time. So again, we can be talking about the same thing in different ways, and I just want to flag that. And so a lot of this work is based off of a hypothesis from a paper by Jeff Hausman, who in 2004 postulated that turbulent diffusivity and water depth can kind of determine regimes under which a diatom will dominate compared to a microcystis. So the idea here is that if you have more mixing in the water column, because diatoms can grow faster, they'll get up into the photic zone and they can outcompete microcystis, compared to if you have really low levels of turbulent mixing, the microcystis is going to be able to swim up, outcompete the diatoms, and you're going to get a harmful algal bloom. And so we were interested in kind of extending this sort of regime level thinking to the delta and start to think about, can we really start to zoom in on the sort of magnitude of turbulent diffusivity and water depths that might favor one species over another. And so the other thing I want to flag in this talk is I'm not going to talk at all about nutrient limitation. And so our hypothesis here is that the delta has such high nutrient levels that they're not actually limiting bloom onset. And so they maybe play more once you start to get really high levels of biomass. But we're just going to assume that for bloom initiation, it really depends on access to the photic zone or light limitation. And so this leads us to the research questions. The first being, how does vertical mixing regulate access to the photic zone for different species of phytoplankton? And my second and sort of a sub question is thinking about how much do we need to think about temperature stratification when we talk about vertical mixing? And does this affect access to the photic zone in a significant way? And for our methods, we conducted a field campaign in the Stockton shipping channel this fall with USGS. And we also ran this one-dimensional vertical water column model. And so I'll talk about both of these today. And so for our field site, we did all of our work in the Stockton shipping channel, which is here. And the characteristics of this channel is that it's freshwater, there's no salinity. It's really weakly tidal, and I'm going to say it's so weakly tidal that it doesn't really affect the sort of vertical mixing profiles at all. Experiences in the summer, diurnal temperature stratification. So every day when the sun starts shining and it gets really hot in the summer, the water at the surface is much warmer than the water at the bed. And then at nighttime, you see that stratification break down. And so you get a constant temperature profile. And of course, this has been well studied, or there's a lot of interest in this as it's a hotspot for HAB events. And so again, a more sort of theoretical before I dig into our field data, but when we think really broadly about how would a daily temperature stratification affect turbulent mixing, I have a little like schematic here. This is my little idealized water column. And you can see that at the top, the water is about the same temperature. This orange line is my theoretical temperature profile at the bottom. And so in the morning, you can imagine this would be a well-mixed temperature profile. And what this means from a physical standpoint is that turbulent mixing could theoretically occur over the whole water column. Or maybe what we would also say is that there's no length scale being imposed by our temperature profile in this example. However, once the day starts going and you start to get the sun is shining at the surface and it's heating up the surface of the water column, this is maybe a more typical temperature profile if you were to take a sample at like 1 p.m. or so, where you have much warmer water at the top and cooler at the bottom, then that does play a big role in the turbulent processes in the water column. And what I mean is that the turbulent mixing to the surface now has this length scale imposed on it. So you can think of it as almost putting a lid on the amount of room that a process could take up or an eddy could physically take up, where the mixed layer depth is now the depth at which a process could happen at the top. And then below the thermocline, you would have another region that theoretically you could have turbulent mixing. And so we wanted to investigate this kind of interplay between stratification and phytoplankton and turbulence. And we conducted this pretty broad field campaign, of which I'm only going to be talking about a subset of the data that we took at Weber Point here, which is that little orange star at the sort of terminus end of the Stockton shipping channel. And I'll talk about, we had a mooring out there for the month of August. And the data I'll be talking about is we had an anemometer. So we took wind data, and we also had a vertical chain of temperature thermistors. So we were able to take temperature profiles at different depths of the water column to compare sort of how that profile changes over time. And we also have this really cool instrument that we were able to borrow called a micro CTD. That I never used this before. I thought it was very fun. And I've shown it here. It's this little orange. It has its little like safety vest on it. but it's this instrument. And what you do is it has this, you attach a weight to it and it lowers down into the bed, and then you yank off the weight and it floats up to the surface. And as it's floating up to the surface, it takes really high profile measurements of both shear, so the actual kind of gradient and velocity, and temperature. And you can use this data to back out interesting parameters about the turbulent processes in the water column. And so an example of what this data looks like, I call every time it's rising to the surface, I call that a cast. So these are 2 casts that we took at Weber Point on August 27th. We went out twice and got samples from this instrument over the course of a day. And you can see here the little purple star on this map and the little green star on the map are the GPS of where the boat was when we took our cast. And then the signal, the shear signal going from the bottom of the cast, the top is the surface of the water column for shear and temperature is shown. And this is just to give you a sense of what this data looks like. And I just wanted to highlight that this little purple signal here really strongly resembles our idealized temperature profile. So you can see here, we have this really clear signal of a mixed layer, that steep thermocline where there's some really strong stratification, and then a layer below. And so what we can do is we can back out an estimate of turbulent diffusivity using this data. And so what I'm showing here, each blue dot is from Turning Basin, which is the little blue star on this map. And each green star or green dot marker is from Weber Point. And I'm showing estimates of turbulent diffusivity over a typical day of August 13th, 2024. So each of those dots is a different cast where we've calculated turbulent diffusivity. And the reason I'm showing both of them is they both actually show a really similar signal, which is that diffusivity is starting off higher in the early morning, and then it's starting to go lower and lower, showing there's less and less mixing occurring as you get into the late morning and early afternoon, and starts to rise back up. And if you spent time in the Central Valley or Stockton in the summer, this is maybe a little counterintuitive because you know that in the afternoon is when you start to get these strong diurnal winds. And so it's weird in a system that receives a lot of its energy from the wind, that as the wind is picking up, we're getting less and less vertical mixing. And we can explain this by looking at what we call the Thorpe scale, or which you can just think of as an estimate. We use this from the micro CTD data of the largest turbulent overturn, or the biggest turbulent scale in the water column. And from this, when we calculate it out, what we see is that that Thorpe scale is going down with the late morning. And what this is a really clear indication that the stratification is the process that's causing our diffusivity to go down. So you see here that that length scale for the turbulent processes is dropping down to like maybe a couple centimeters or nearly 0 in the late morning and early afternoon as that stratification is really strong. And so we're showing here this really big difference that stratification is making and reducing the amount of vertical mixing that's happening in this channel. And for the second part of this study, it's obviously really fun to go out and collect all this data, but it's really expensive. And so one hope we have is that, it's really nice if you can get a diagnostic or prognostic model running that you can use to sort of test out different scenarios and explore other questions. And so we have this really simple one-dimensional water column model that takes in wind and temperature and tide as its forcings. And I can talk about some of the details on it if anyone's curious. But the output of this is you can get estimates of diffusivity and dissipation and length scales. And the thing that was really fun is that we were able to use our mooring data to force this model. And so instead of asking about some idealized situation, we can use this model to try as best as we can to simulate a day in August at Weber Point in the Stockton shipping channel. And so we can take our wind data at the top and force the actual temperature field that we measured. So I'm imposing a temperature field, and we can run it for any day and look at how it performs compared to some data we collected. And the other thing we can do is then we can add phytoplankton to this model. And so we can impose phytoplankton using an advection diffusion equation, and then have some swimming, motility, and then look at different growth and loss parameters and for different species of phytoplankton and how they might perform in some day in the life at the Stockton shipping channel. And so this is my validation plot. I was pretty happy with this just because these things, it's a little bit like comparing apples and oranges, and that we have this simplified 1D water column model and then this like noisy high frequency micro CTD data that we've calculated a diffusivity estimate from. But what you can see here is this is a validation of our model diffusivity compared to our observed or calculated diffusivity. So each of these green dots here is the turbulent diffusivity from a micro CTD cast. And then the blue line, the faded one, is a higher frequency signal from our model. And the darker blue line is a smooth or moving average from our model. And the main thing I want to say is that we're able to capture this same signal of it dropping during the day as that stratification is kicking up. And to us, this is saying that, the physics in this channel are pretty simple. We have wind at the surface, you have heating at the surface. then this is saying that you're able to kind of capture these physics reasonably well with a one-dimensional model, which is nice because these run really fast, like in a minute. And so I'll close by talking about a simulation we ran, curious about the conditions of how much does stratification actually matter if you're a diatom. And so we ran two simulations. The first is our stratified simulation. So this is where we take our temperature field from our thermistor chain from Weber point and impose it. And so these diatoms in this simulation are experiencing the realistic temperature field that happened when we were out in the field. So they're experiencing stratification during the day and then kind of mixed conditions at night. And then I have this other theoretical simulation where I just impose a constant temperature field. So I just took the average at every time step and imposed that as just a constant line. And this is asking the theoretical question of like, if there wasn't any stratification in the Stockton shipping channel, what would have happened from a biological standpoint? And are these two significantly different? And so I'm show a movie and there's going to be a lot happening. So I'll walk through the pieces of it so you can better absorb what happens when I press go. So in the top left-hand panel here, I have our light data. This is from one of the CIMMYS regional monitoring stations. And so this is photosynthetically available radiation. And so this is just showing us when the sun is shining. When it's 0, it's nighttime. And when the sun is shining, it's obviously quite high. On the second panel, we have wind from our anemonitor data. So this was used to force our model with, and it's really noisy. So I just plotted a smoother line in the dark blue. And then at the bottom is this fancy calculation called the potential energy anomaly. And this is just a measure of stratification. So you can think of this as when this pink line is really high, that means the water column is really stratified. And you can see at night, it drops down to about 0. And then on the right-hand side is kind of going to be the key thing, which is our diatom concentration. So there's going to be a blue dotted line that's going to be the diatom concentration as a function of depth, where 0 is the surface and -6 is the base of the channel. And the unstratified will be the blue dots, and the stratified simulation, those diatoms will be shown as this green line. Okay, so now we're starting off. And so again, the thing that I want to note here is during the day, what you can see is at the very surface, our stratified simulation, the diatoms are benefited by that stratification. But because of the higher mixing that unstratified simulation has, it's able to better assimilate those growth into the whole water column. And you actually are going to get a lot more biomass growth in the unstratified simulation, just because that mixing really helps if you're a diatom and you sink naturally. We can watch it for a day or two. There's a lot going on. But anyway, you can see kind of they both respire at night. So again, this is during the day. We're seeing the late afternoon. But over the course of the day, and if I were to run this for the whole month, what you'd see is that the unstratified simulation starts to build up significantly more algal concentration. And this is kind of showing us that the stratification in this channel is hurting diatoms. And so it's going to benefit microcystis and hurt diatoms. And I'll go to my next slide with an interest in time, which is that the kind of conclusion I'm going to have is that the turbulent diffusivity, if you actually look at the output from our model, from our unstratified simulation and our stratified simulation, this is turbulent diffusivity plotted here. That you can see that it's about 100 times stronger. This is on a log axis during the day when the water column is unstratified. So when we think about vertical mixing and we want to care about it, this makes a really big difference, these physical processes, and it can make a huge difference in the output of biomass from different species, especially depending on their motility and growth rates. And so in conclusion, I just want to emphasize that temperature stratification in these dead-end channels, like the Stockton shipping channel, is probably reducing turbulent mixing by an order of magnitude on a typical summer day. And we could dig into that more if we wanted to play around with the 1D model. And that our numerical simulations, one, that we're able to replicate the physics of this channel fairly well. And so that's nice in terms of thinking, okay, this is a tool we could use to answer other research questions. And it's showing us that unstratified conditions are better for diatom growth. And so if we wanted to dig a little farther, we could start to play around with some of those actual growth and loss parameters. And that's some of the future work we hope to do. And we're also hoping to include species competition with microcystis. And I do think at that point, we might have to think a little more about nutrient limitations if we want to think about what causes an actual microcystis bloom to develop. But I'll close there. I also want to acknowledge our funding from the State Water Board Division of Water Rights. And then we had a lot of help out in the field. I also really want to thank the USGS biogeochemical field team. They were a delight to be out in the water with and helped a ton. So thank you so much.
Thank you so much, Sienna. Sorry for getting that detail wrong again. I have my cheat sheet back. So yeah, but we have a few minutes for questions if anyone in the room. I think I see a hand over here.
Hi, great talk, thank you. I was wondering if you could speak to how big of a temperature difference you need before stratification starts to really affect vertical mixing.
That's a great question. The problem is it's a little bit nonlinear because it also, the thing that matters is how much the wind is blowing. So these two things in combination really matter in terms of setting whether or not the wind, sometimes the wind is able to mix out that stratification gradient. So they're kind of competing with each other at any given point. In this, it was just a couple, it was like on the order of 1 or 2 degrees. So kind of slight, but yeah, it made a difference.
I was wondering if you could speak a little bit, kind of expand on what you got towards the end about differences in growth rates between the different genera of diatoms and other organisms, and kind of where those growth rates generally come from, and whether, is it, kind of pure culture lab measurements, and then if you just had a sense for what the existing knowledge of differences in growth rate between different organisms, even within the same group like diatoms, could be.
Yeah, that's a great question. So the estimates that we use for this study came from Jeff Hausman's paper, but in general, yeah, these do come from literature. And I honestly, it's something I struggle with in research is, whenever you're trying to parameterize a biological process, it just feels like such a crazy simplification when you have this exponential function where you're just plugging in a parameter. So we've been trying to think for the next stage of the study, how to go about thoughtfully, if we want to actually think about, okay, what would cause microcystis to grow? What's a realistic growth rate? And the nice thing is this model runs so fast, I think we're more hoping to do like a regime style analysis where it's like, okay, if these are the conditions, if the growth rate's anywhere between like, 0.2 and 0.7, you're going to see a bloom, as opposed to trying to get maybe a literature best possible estimate. Because again, it's such a simplification. In terms of the difference between microcystis and diatoms, I think it's about an order of magnitude for the growth rate. And it's one of those growth rates, it's like 1 over time is the unit. Yeah.
I kind of have a 2-part question. One is, I believe this Stockton shipping channel is still used for shipping. Was there any indication of like mixing due to these giant vessels? And then a second question I have is the Stockton shipping channel representative of most of the dead end sloughs in the Delta, or is it sort of unique in the size of the channel?
That's okay. I'll answer the second one first and then the first one. I think that's a really interesting question that when we think about HABs in the Delta, we'll have to really zoom in on, which is thinking about, and it's one of the management questions the workshop talks about today, but when you think about how similar are certain regions of the Delta and how do you define like similar? Does it, average velocity or average diffusivity or average wind? And so for now, I would say that this feels kind of safe to say this is like a prototypical channel, but in terms of, no 2 channels are the same and locally things can look pretty different. And so I think that's a question that, yeah, requires quite a bit of future research and thought. In terms of, so the ships actually don't come all the way down. So they come to that turning base and that's where they turn around and go out. And in terms of like mechanical, the mixing of the ships, I think that's a really interesting question. And I don't think anyone's thought about that. I would think that it's so intermittent that it's probably hard to think that it makes a difference like on kind of a longer time scale. But I think there are studies they've done, I want to say in like Puget Sound, where they're interested in like sediment kick up from like ferries and stuff. So there is interesting thought about that.
Hi, great talk. Your animation of the diatom growth with the mixing strength was really cool. And it got me thinking of if there's been any thought given to identifying maybe some threshold mixing, or stratification strengths that might favor microcystis growth in like a similar sort of analysis.
Yeah, no, thanks. I think that would be really interesting. And so, yeah, whenever you think about a regime plot, it's like, okay, what are we going to make these axes? And so I think right now I'm leaning towards that. I think that some sort of measure of stratification would be a great axis to be on, especially given what a difference it made in the diatoms And future work, I really am curious with the microcyst if it'll have that same sort of like, oh, it's 20% more with, you know, stratification. So, yeah.
In your future work, you mentioned looking at nutrient competition with microcystis. Would you also look at light competition? Like, would they shade it out?
Yeah, so there is self-shading in this model. It's actually, it really slows the model down because you have to like integrate at every time step. So yes, and that's actually the Jeff Hausman paper. The idea of it is competition for light. And so there is self-shading. I think one thing that also, again, biological processes, hard to model because you're simplifying. When microcystis starts to form those surface mats, it feels like that's a very different sort of self-shading than when you have the little diatoms or little microcystis dispersed in the water column. And so, yeah, we're hoping to think about like explicit competition between species and just want to be careful about the way we represent the self-shading once those microcystis concentrations get super high.
Fascinating talk. Thank you.
Thank you so much.
Yeah, thank you, Sienna. That was wonderful. Our next presenter I want to invite up is Nick Framstead, who is with the USGS Biochemistry Group and is interested in ecosystem ecology and controls on primary productivity in aquatic systems. Let me transfer here. All right, take it away, Nick.
Thanks for everybody for sticking in there for the last talk of the day. Yeah, I'm Nick Framstead of the USGS Biogeochemistry Group in California Water Science Center. Today, we're going to be talking about drivers of phytoplankton dynamics in the South Delta, south of Stockton, like the South Delta. So we'll be getting into sort of a new region for today. And this is in collaboration with our collaborators at the DWR North Central Regional Office. So the study takes place in the region outlined in the rectangle here. So this is an important region that encompasses the State Water Project facilities and the Central Valley Project facilities as well. It's characterized by shallow, narrow channels, and flows are regulated by the San Joaquin River flows from the south, tidal excursions, local agriculture withdrawals in some of the dead-end sloughs, as well as temporary rock barriers that are placed seasonally within this region to maintain water elevations. And then, of course, the exports facilities. Past studies in this region have focused mainly on salinity and dissolved oxygen, but we're going to dive in more to the phytoplankton dynamics. So the objectives were to identify the status and trends in phytoplankton and cyanobacteria harmful algal bloom or CHAB dynamics in this region, and then identify potential drivers of CHABs. So zooming into the South Delta, this is a rainbow map of all of the stations in the Delta. There's a lot of data collected here by a lot of different agencies. And here we have them split up regionally by some of the different water bodies in this region. And there's a lot of data. And there's also, I'll draw your attention to some of these yellow block boxes. These are the temporary rock barriers placed May through November to maintain upstream water elevations for local ag users. So traditionally, or I guess naturally, flows come from the south, from the San Joaquin, and work their way down the San Joaquin or up Old and Middle Rivers to the confluence with the Sacramento River and out to San Francisco Bay. But sometimes there are weird instances where during low flow years from the San Joaquin, where we actually get reverse flows, which are indicated by the red arrows, where Sacramento River water is pulled south through the Delta towards the export pumps through Old and Middle River, through Victoria Canal and Middle River, and San Joaquin flows that are pulled westward to the pumping facilities. So it's quite a hydrodynamically complex region, and this has interesting implications for phytoplankton dynamics. And just to give you an idea of what these stations look like, they can be pretty narrow channels. And here is station ODM, which is right adjacent to one of those rock barriers, and you can see a backhoe on the barrier. I'm pretty sure this is in the winter, so it might be in the process of removing a barrier. We also see a lot of aquatic vegetation, and it's an important aspect of this region. Decent amount of FAV can clog up these channels, which has implications for light availability. So just setting the scene, nutrients tend to be replete in general in this region. So here we have dissolved inorganic nitrogen on the plots to the left, and these are all of the regions. And on the red line is indicating literature values that indicate potentially limiting nitrogen concentrations. All of our values are pretty clearly above that line. Nitrogen is not really considered to be a limiting factor. And then on the right, I have phosphate plotted, and phosphorus is also generally not limiting. So there's plenty of nutrients available for primary producers. There's also warm temperatures. So temperature is also a very important variable in determining phytoplankton productivity and bloom, probability of having a bloom. So the red lines denote the 19 degrees Celsius mark, which is Peggy Lehman's threshold above which microcystin tends to do really well. So during the summer, we're pretty much always above 19 degrees Celsius. And then in a few regions, Middle River and Old Middle River and Clifton Court Forebay, we're above 25 degrees Celsius for our medians temperatures in July and August. So it's a very warm region as well, likely a result of being so shallow and having higher residence times. So seasonal patterns and discrete chlorophyll, we see that there's July, August blooms that occur regularly in all of the regions. We also have a smaller local maximum in February and March, but this is dwarfed by the summer blooms. We have generally high chlorophyll concentrations, up to 60 micrograms per liter. And the line that I have plotted, the red line, is indicating the 20 microgram per liter threshold above which we're considering a bloom. So we regularly have blooms in some of these regions. Yeah. And it kind of as an aside, we are looking into long-term discrete chlorophyll dynamics. In our upstream-most station, C10A, is at the San Joaquin River near Vernalis. We see a consistent decrease below the long-term average, which is plotted as this black line. So each bar represents an annual mean chlorophyll value. Blue lines are positive deviations from the annual mean or the long-term mean. Red bars are negative deviations. So we can see that since 2016, we've been seeing negative anomalies from the long-term mean, and these are reflected also in the stations immediately downstream at MRU, OH1, and TWA. We're still trying to do some change point analyses to identify the date, the exact time that this change point occurred, and if this negative trend after that change point is actually significant, and as well as trying to figure out possible management actions or changes to this region that might have precipitated this decline in chlorophyll. But anyway, moving back on to the phytoplankton dynamics. So given the nutrient concentrations and the warm temperatures, we thought we would see a lot of potentially toxic cyanobacteria dominating the phytoplankton community. And so let me orient you to this plot. This is relative biovolume at each of our stations that we have data for. Some of them only collected data in the summer, so you see some gaps in between the bars. And the potentially toxigenic cyanobacteria indicated by the red include Microcystis, Aphanizomenon, and Dolichospermum taxa. So at some periods of time, we do see that these toxigenic species are dominating the community, notably at stations C9, MHO, and ORM. These are more interior to the South Delta, near to Clifton Court. However, when we look at the chlorophyll A concentrations that were paired with these biovolume samples, we don't see the same story. So these axes vary, the y-axis varies for each station. So C9 only goes up to 10 micrograms per liter, but C10A goes up to 40. So whenever we see large chlorophyll events, we usually see the community dominated by both centric diatoms, pinnate diatoms, and sometimes green algae. However, whenever we see the community dominated by potentially toxigenic cyanobacteria, they're usually present at coinciding with low chlorophyll A concentrations, indicating we probably didn't see a significant bloom event of these taxa. There's some assumptions in these data that biovolume doesn't correlate directly to chlorophyll A content. It's not constant across all taxa, but we would expect to see higher chlorophyll levels associated with those events if there was a CHAB. And this is an NMDS plot of all the phytoplankton community data. So closer together points share more similarities between their phytoplankton communities. We see that the potentially toxigenic cyanobacteria tend to group up here in this corner, but there's a lot of overlap between communities, and this isn't judging by the length of this P-tox vector here. It's not driving a ton of variability in the general community dynamics. However, we do see a lot more black points up in this region that are dominated by P-tox taxa, and those correspond to Clifton Court forebay inlet stations. So these are, let me orient you. This is station C9, which is an EMP station. We also see that San Joaquin River stations here in the yellow are maybe grouping a little further away from the P-tox, meaning they're less dominated by potentially toxic cyanobacteria. However, there's still a lot of overlap, and there's not a lot of differences in community between the different regions, which is interesting. However, there is a bias between these two stations because EMP collects data monthly. The other stations are just collecting summer samples, so you'll just see a lot more data points from those stations in this plot. So moving on from the whole community, we're just going to zero in on microcystis. So we're going to talk about microcystis visual index scores, which are a semi-quantitative measure of microcystis abundance, with 1 being absent, 5 being very high abundance. And here we grouped samples by water year types, and we have this plotted out for each region of the South Delta and the percent of samples that fell within each microcystis visual index score. So here we see mostly microcystis being absent or present at low concentrations, indicated by the blue bars and the purple. However, we do see some high abundance of microcystis during mostly critical and dry years. However, these are still at low percentages of samples during those years. Some exceptions are at Victoria Canal and Clifton Court forebay inlet stations, where we see microcystis abundant even in below normal or wet years, which is an interesting pattern. But again, microcystis visual index rarely exceeded a moderate rating of 3 in this region, which is kind of surprising. Looking at cyanotoxins, only microcystin has been detected in the South Delta, and there's been no detections during wet years. And concentrations were always below the EPA recreational public health limit of 8 nanograms per liter. And we see most of our detections in the critical or dry years, indicated by the yellow and red lines. So looking at potential drivers. So this is a picture of a microcystis bloom, also some aphanizomenon sprinkled in there. This was a picture taken in Victoria Canal in July 2018. And in case you were curious, these were the associated chlorophyll, continuous chlorophyll fluorescence, and visual index scores. So it's given a moderate score of microcystis abundance. So we wanted to look at water speed because, as Sienna beautifully set up, turbulent mixing is a really important determinant of microcystis bloom formation. With greater water speeds, we have lower residence times, so it's a proxy for water residence time. We do see that we get higher continuous chlorophyll values associated with slower water speeds. And there's somewhat of a threshold of, we see these higher chlorophyll concentrations tending to occur more often below about 1/4 meter per second water speed. And again, we're talking about water speed, not velocity, because in our, the directionality of flow, whether it's positive or negative, doesn't matter. It's just whether water is moving and creating turbulent mixing or not, and reducing residence times. So looking at the visual index scores, here's our density plots of microcystis visual index for each rating. So we have different regions showing different patterns. We see that higher microcystis visual index scores are more associated with slower velocities, and lower microcystis visual index, where microcystis is absent, is more common at higher water speeds. However, there are a couple exceptions. Again, Victoria Canal and Clifton Court forebay inlet station or regions show a different pattern where we see even high microcystis abundance even at higher water speeds. And this may be due to the export pumps being nearby, these pumping facilities. So these regions here are the ones showing high microcystis even at higher water speeds. They're located nearest the export pumping facilities, and we might be importing blooms from this central delta as they flow southward due to the export pumping, drawing water from those regions. So that might explain this anomaly, but there are other causes that could be possible as well. So here are the results of an ordinal regression on microcystis abundance. Because this is microcystis visual index is a semi-qualitative measure, we bin them to be more robust. So we have absent, low, and high categories. And the regression temperature and prior week water speed came out as significant covariates in the regression. And as you can see, at higher temperatures, we have a higher probability of detecting microcystis at both low and high concentrations. And at low temperatures, we have a higher probability of microcystis being absent. So this is what we'd expect. And we see the exact opposite relationship with prior week water speed, where at lower prior week water speeds, we expect we have higher probabilities of detecting microcystis, both at low and high abundances. And as speed increases, we're more likely to have microcystis be absent. So this was an interesting pattern, and it did fit what we expected. However, the probability of detecting high microcystis, according to the data from this region, is still relatively low. Again, microcystis blooms don't seem to be a common occurrence in this region. So in conclusion, the South Delta has few detected CHAB events, with high chlorophyll blooms dominated mostly by diatoms and green algae. There's potential long-term decline in discrete chlorophyll A concentrations at the San Joaquin station and those immediately downstream. However, we're still investigating why that might be the case. Water temperature and water speed are both found to significantly affect microcystis abundance, with higher temperatures and slower water speeds favoring higher microcystis abundances. And there's a need for further analysis to develop threshold velocity estimates that may prevent microcystis blooms and growth in this region. And this would be a region-specific parameter. And understanding cyanobacteria blooms in this region is critical for supporting state and Central Valley Water Project operations and for local agricultural uses. With that, I'd like to thank our funders from the Department of Water Resources. Special thanks to all of the agencies that collect quality data in this region, as well as NCRO staff especially. And we'd like to thank Eli Atelyevich, Steve San Julian, and Brianne Sakata for helpful input and data into this project. Thank you. I'll open it for questions.
I think we have time for about one question if we have. Okay. Or we can expand it, yeah, if people have more.
How do you think microcystis blooms will change in the future?
If the precipitation levels continue to be more variable and we have more frequent periods where San Joaquin River flows are lower and project exports are outmatching the inflows, yeah, that could definitely create lower velocities in this region, increase the frequency of null zones, and that could potentially favor microcystis and increase temperatures. So yeah.
All right, if that's it, another big thank you for the presenters of this panel. And I think with that, we're ready for closing remarks.
Yeah, we just want to thank you all so much for coming out again and sticking here to the end, and those online for tuning in. I think this was a great workshop, some much needed FaceTime with our colleagues and cool ideas exchanged. This ends this year's workshop, but be better if I'd actually organize things. My brain is fried. Thank you all for coming. The early career presentation and poster awards are going to be announced via email. The other thing to announce is that Adam has been my excellent partner in crime in planning this workshop. And next workshop, I'm stepping back. And so Adam will need a new partner in crime. So if you're interested in helping to plan next year's workshop, let us know. Anything else, Adam or Stephanie?
No, I think that's it. Thanks again, everybody, for coming. Be sure and grab your poster. And like Rosie said, I'm looking for a co-organizer next year. So keep that in mind and be thinking about your presentations next year too. Thanks, everyone.